So, welcome everyone to the launch of the Immuno-Pathology Network, which is being set up by the Department of Immunology and Information at Imperial College. So we started planning this actually quite a long time ago. We had a meeting in December the 10th, 2019, with a sort of steering committee that has most of these, or all of these people on, just a few extra ones. And we then, at that meeting, decided what we wanted to achieve at the network, and I'm going to be telling you about that in the next couple of slides. And that my friend and colleague over there, Guido, has been sort of heavily involved with me in organising, setting up the network since then. So what we're trying to achieve with this the network is to facilitate interactions between scientists and clinicians. Initially, the idea was to do that between researchers at Imperial College, but then we decided that it would be advantageous and interesting to expand the interactions to include the Francis Crick Institute, the Institute of Cancer Research, and the Wellcome Institute, where each of those institutes have significant interests in immunology-related questions. And the network is also planning to promote collaborations between Imperial groups and these partner institutes. So the research areas that we're sort of going to be focusing on in the network are listed here, which is pretty much everything. Inflammatory diseases, infectious diseases, vascular diseases, lung diseases, brain diseases, digestive diseases, and cancer. And then also, more generally, immunology, immunogenetics, but also specifically taking advantage of the sorts of work that get done at Imperial. We're also involving bioengineers, chemists, and also people from computing working on artificial intelligence and machine learning, and that type of activity. So it's very broad, and there's been a lot of interest in setting this up. I think there are over 150 groups from Imperial that have expressed an interest, 28 groups from the Crick, over 10 groups from the ICR, and 5 groups from Sanger. So how we're going to be sort of running the network is via a dedicated workshop, which you can find at our departmental page in the research section. And this has been set up with the help, or not the help, she basically did all of it, by Genevieve Timmins, who's our communication manager, and she's done a fantastic job in setting up a very nice website that has all the information that you need to sort of find out who's involved and what sorts of activities are being worked on. So obviously one of the main things is what areas of research we work on. So for each research area, we have a dedicated page. So I've shown here the brain diseases, and you've got a nice picture of a brain, just in case you're not sure which page you're on. And also, you can also see each of the members who are working in this area with their picture and a link through to their home page, so you can find out more about those people if you want to. We're also going to be highlighting particularly interesting pieces of work that have been done by network groups in the network highlights section. And I've just shown one of them here. This is one by Gad Frankel, who's recently published an omics paper looking at Citrobacter infection and the role of type 3 secretion systems in science. And and these network highlights will have a sort of short summary of what the main findings of the work is, and a schematic so you can get an idea of what's going on. And then obviously you can look in more detail at the primary publication if you want to. And One of the other activities we're planning, and this was particularly championed by James Harker and Marga Dominguez-Villar, who are going to be running this, is we're going to be having journal clubs. We're going to run them once per month on the third Thursday of the month, and they're going to be run on Zoom. And the format will be that someone will be invited to present their latest findings, and they give a short presentation. Then the people who turn up can then discuss what they think about the work in breakout rooms, and then there's a general discussion. And the target audience here is early career researchers, postdocs, and students. And one nice thing about this is it will allow students at Imperial to meet students that are working at the Crick or the Sanger or the ICR and form their own mini networks that may be useful for their research in the future. And then the last activity that we're going to be running from the network is workshops. We decided that was the format that would be most useful, and we're running one of these per term. And the workshops are going to be taking place at the Francis Crick Institute, because that is the easiest place for all of the members of the network to get to, including the Sanger, ICR, and obviously the Crick. And also they've got good facilities here. So the first workshop is on March the 4th, and it's on cancer immunology. And that's being organized by Jessica Strid and Alan Melchers. And you can see the speakers have already been chosen there and confirmed. So hopefully some people will be turning up to that. That'll be working both in person at the Crick, but also you'll be able to attend that online. And then we've got two more workshops scheduled for next year. The first of these, or the second workshop, is on inflammatory diseases, and that's on June 17th. And that's being organized by James Harker and Emma Davenport from the Sanger. And then the third workshop is on infectious diseases, and that's going to take place on September the 23rd, and that's being organized by Patricia Barral and Teresa Thurston. And finally, there's a page on the website that allows you to join the network. So if you're interested, if you haven't already done this, if you're interested in taking part in the network, you need to sign up and maybe encourage people in your lab to sign up to this as well. And then you will then receive information by email about what's going on. And one of the other things that was decided at the meeting we had back in December in 2019 is that we wanted the network to facilitate and promote interactions between Imperial and pharmaceutical companies. So we've invited to this meeting representatives from GlaxoSmithKline, AstraZeneca, Sanofi, and Novartis. And each of those companies are going to give short presentations which sort of give an overview of the sorts of things that they're interested in from a drug development point of view. So obviously some of the stuff that's going to come out of the network is potentially translatable, and through the symposia, these companies will get access to or hear about new stuff coming out of the network, and then maybe follow that up by setting up collaborations. Now, if you're at Imperial and you want to set up a collaboration, you have to do this with the industry partnerships and commercialization team. And there are two people that are key people that you would need to talk to: Stephanie Morris and Vaira Musavi. And hopefully the network will be end up resulting in new collaborations with these companies, and in the long term, hopefully development of new drugs. And finally, I'd like to thank the sponsors of this meeting that have enabled it to happen and are paying for the food and the booze. And so you've got each of the pharmaceutical companies that I've just mentioned, LifeArc, the Imperial NIHR Biomedical Research Centre, the ICR, and also Becton Dickinson, Miltenny, and Queergen, who each have tables outside and if we'd like you to go and have a look at what they are selling during the course of the day. Okay, so that's it, and we're now going to hear a series of talks during the day that is going to give you a real overview of the breadth of immunology-related research that is going on at Imperial College and the partner institutes. And we're going to start off with Marina Botto, who's the head of the Department of Immunology and Inflammation, and my line manager as well. Right, take it away.
That's not a good start. How do you get out from your talk? All right, a second. Do I have a pointer here?
I think you can use the computer. There isn't a...
There's no pointer. Okay, I'm with her. Right. So, well, I will start really with saying my sincere thanks to the organizer for getting all of us here, and in particular to Steve, who has been really the driving force behind this network. And I have to be saying, nothing would have happened unless exactly he was so determined to make sure that this network was happening, despite exactly the difficulties on the way. And as Steve has said, this is something that started quite a few years ago. And it's a great pleasure to be here today and actually seeing quite a few people in person after such a long time of online meetings. So I'm just going today giving you really a brief overview of the Department of Immunology and Inflammation that I have the honor to lead. And clearly, I want to start with some apologies, and apologies to the member of my department, because in the next 10 minutes, I won't be able to give really justice to all the work that they're doing, and certainly to the cutting edge activity that they clearly produce every day. So I'm starting to say sorry. It will be a very generic overview of the topic of research and not exactly any details. But clearly the meeting is supposed to give you the opportunity to talk with them and to discover more about what they're doing. The Department of Immunology and Inflammation, like most of the department in Imperial College, is really a mixture of basic science and clinicians. And clearly, we wanted to integrate discovery science with the clinical medicine. And in the case of our department, inflammation and immunology are the basis of the diseases that we are interested in. They are mainly focused on hematology and kidney diseases. And if every time people ask me, what does your department differ from the Department of Infection Diseases that you hear later on, I tend to respond, we are looking at sterile inflammation, or at least in many of the diseases we are interested in, at least the pathogen is not known. But the key point is that in our department, we just have a structure where basic science, it's really fully integrated with the clinical aspect. And as you see, the department has got 3 centers, the Center of Hematology, Center for Inflammatory Diseases, Center for Molecular Immunology, Inflammation. But integrated in this structure is that these 3 centers are very much part also of a clinical delivery, which is clearly done through center like the BRC, which is our Imperial Biomedical Research Center, and the Imperial College Trust, which is also part of a bigger picture, which is the HSC, so the Imperial College Academic Health Science Center with 7 other partners. So when you look at this department, you will find it very difficult to, or at least you will find many times that the same person sitting in all this structure, which means there is a full integration of the basic science that happens in the laboratory to the clinical delivery in the trust. So let's start to give you an overview of what's going on. And I'm starting with a Center of Hematology. And the Center of Hematology, which is co-director Professor Karadimitris and Professor Lo Celso, they are really working on three main areas. But again, just keep in mind, I'm giving you only a flavor. There is an hemato-oncology with a clearly great interest in acute chronic leukemia like CML, and in acute leukemia, you can see Cristina Lo Celso interest in the microenvironment and the interaction of the bone marrow using multiphoton microscopy, where she's looking at the aspect. I wish I had the pointer. No. And the multi-aspect of perfect. Thank you. Yeah, you have an interaction between the cancer cells and the bone marrow niche. In the center, there is a great interest in the multiple myeloma. We have the Hugh Jocelyn Langmuir Myeloma Center that will be open next year, where Professor Karadimitris and many other components of the center are working on addressing this very important disease. And they're addressing from the genomic center, looking at regulatory genomics of the myeloma, as well as looking at the metabolism in myeloma, in multiple myeloma cells. For example, Olga Owner, which is also associated with the Crick, is looking at the metabolism of the cells, targeting in particular the GCN2 inhibitor as a potential target. Another important area of interest in the center that is unique is the developing of cell therapy, and in particular, Professor Karadimitris is looking at the invariant NKT cells as a platform for immunotherapy in blood cancer, with the advantage of using this cell therapy, which got the advantage of being possibly much more effective in killing the cells, and really off-the-shelf therapy that will not need the expansion of the cells from the patient. So for what I read, the next part, slower computer. The other two topics of the department are around the coagulation or thrombosis of homeostasis. There is a long-standing interest in this area, a group led by Jim Crowley and many other members of the department. And again, this is an area of great interest at the moment. If you think at one of the main consequences of the coronavirus, we have seen clearly the problem of the thrombosis. And there is a long-standing interest in Adams 13 and von Willebrand factor. And the department is developing different approaches to try to target a different component of the coagulation, with the developing of new antibodies that could be targeted for deep vein thrombosis, as well as ischemic stroke, and the new novel anticoagulant therapy. So there is a long history of interest in the thrombosis. Similar, there are also interesting two, let's say, more rare situations. One is a red cells condition called the diamond black fan anemia, which we have clearly the National Center for this rare patient. And again, the interest is looking at the difference in the patient, in erythropoiesis in this patient, which have this quite severe condition. As well as there is a great interest in autoimmune cytopenia, specifically thrombocytopenia, led by Dr. Nicola Cooper, who has probably collected a large collection of autoimmune thrombocytopenia in the country. So as you can see, the department of the Center of Hematology has got this general interest in this area. The Center of Molecular Immunology and Inflammation, which is led by the two co-organizers of this meeting today, which is Steve Lay and Guido, are clearly looking at much more molecular basic science aspect of the disease. And Guido is clearly well known for his interest and work on FKB, and now is looking at targeting NF-κB in the context of multiple myeloma and diffuse large B-cell lymphoma. And there is an ongoing clinical trial at the moment with a compound called the DTP3, which is exactly being developed following his basic science, as well as interest in cancer metabolism and looking at the development of therapy in the context of colon carcinoma and ovarian cancer. Steve is clearly also widely internationally known for his work on TPL2 and CARD14 signaling, and is now interested to develop clearly anti-inflammatory cancer drug targets to the TPL2 MAP3 kinases, which looking at the role of this signaling in IBD and immunoresponse to bacteria, as well as his interest in CARD14 psoriasis. And then the well-known is Danny Altman with his work on T-cell subsets and antigen response to virus, and is become very known in the context of coronavirus. I'm conscious of the time because Guido is standing up already. Finally is the Center for Inflammatory Disease, which is really focusing on various conditions around probably autoimmunity and the kidney disease. So we have a very long-standing interest in lupus, and we cover every aspect of lupus, from the genetic studies led by Dr. Jim Peters in collaboration with Emma at the Sanger, to molecular mechanism, to murine model, to clinical trial. And similar, we have in ANCA vasculitis a long-standing interest in this work, again from basic science to clinical trial. We are sitting in the largest renal unit in the country, probably in Europe, and the key topic of interest is renal transplant with antibody-mediated transplant rejection, and we are trying to develop new diagnostic tools that goes beyond the old style of looking under the microscope. Can we develop something in a signature, transcriptomic signature that will add to the eye of the histopathology? And again, we have a very niche interest in complement biology from Professor Matthew Pickering and myself, and interest in some probably rare but unique conditions that are driven by complement involvement. In particular, at Hammersmith, we have really developed a new disease that's called C3 glomerulonephropathy, and clearly there is a role of complement also in IgA nephropathy, and there are clinical trials at the moment involved in targeting this complement in this disease. Finally, we have a basic science, because as I said at the beginning, this is an integration of basic science and clinical work. And the basic science goes from looking at lymphoid stress surveillance in the skin, and Jessica Street has been mentioned before for her interest in skin cancer. We're looking at cell metabolism and autoimmunity, as well as Alice Denton is looking at the role of the stroma and lymph node and response and vaccine and complement biology. Finally, is just joined us Peter Brodin from the Karolinska Institute, and he's well known for his approach to looking at the development of the immune system in newborn. And Peter will develop essentially a very multi-omics approach to look at how the variation in the immune response determine our life, predict our response to the stimuli that we encounter during our life. And he's about to set up his own laboratory in the next year with us. And finally, but not clearly not just because it's less important, there is also the molecular inflammation that we are looking at with Dr. David Thomas, who has discovered a new component of the reactive oxygen generation, which is called EROS, and we are interested clearly in mechanism of tissue damage, a role of the macrophages. So I just want to finish. I'm sorry, it's been a very wide and broad brush overview of what we're doing, and I just want to thank all the people in the department who are actually doing the work and are driving all this topic. And as I say, I apologize for the many names that I have not mentioned, but because it's absolutely restrained, because Guido is sitting in front of me now, just telling me to stop. So thank you.
Thank you, Marina, for a very dense topic. I have a question for time for the question.
I don't think it will be.
If not, we can move to the next speaker. Thanks, Marina. So the next speaker is Professor Peter Openshaw. Can you hear me? Peter Openshaw from the National Heart and Lung Institute at Imperial College London. Peter will tell us about immune responses to respiratory viruses. Right place.
Do you need the pointer?
I think I can point from the computer.
While Peter is setting up, as a cue to the speaker, we have a busy schedule, so we'll be standing up after the 10 minutes.
Great. Okay, I just hold on a second. Yes. I feel like rather like Boris losing his notes. Okay. So it's a great pleasure to be here, and I just was wondering how possibly to tell you a story that hangs together in the few minutes that we have to give an overview. And I thought that this title of, right place, right time was a good one, because I think, we have been sequentially lucky in being in the right place and the right time in terms of working on respiratory viruses. But I think equally, it's so important to think about place and time in terms of the immune responses. So the focus on the mucosa is very much what we've done for many years. But of course, we have to use the peripheral blood to sample. So let's just go through a bit of what, the story of being in the right place and the right time. So this is the 2009 influenza pandemic, the swine flu pandemic, when the Wellcome asked us to coordinate a national project called the Mosaic study. And this was 45 co-investigators, all our friends from all around the country, people who could sign up immediately to try to discover the immunology, the pathogenesis of H1N1 flu when it first hit our shores. And it was an immense effort setting this up very quickly, but immensely rewarding, I think, in trying to produce a new atmosphere of openness and collaboration between all of us, focused on this subject that was of such national and international importance at the time. It was the basis of the study was to do a very, very intensive deep dive into the sampling and clinical characterization of patients that were admitted to hospitals in the UK. And because of funding limitations, we ended up just focusing on hospitals in Liverpool and in London. So we had 11 hospitals. We managed ultimately through 3 waves of the H1N1 to get 255 patients and a vast amount of clinical information and a very large sample bank. And this allowed us to really collaborate widely and try to characterize what was going on in terms of pathogenesis. And this gives an example of looking in the serum, the nasopharyngeal aspirate and the nasabsorption samples. So these are absorbative strips that were developed by Trevor Hansel over many years of work in order to sample nasal fluid very atraumatically And it's an extremely useful technique for finding out what's happening in the anterior compartment of the nose. And you can see in this particular example, we've got IL-1 beta, we've got IL-6, and we've got IL-8. And I think, a remarkable finding is that even in healthy controls, we've got a lot of IL-8 in the anterior compartment of the nose, but that generally, as severity increases, so a lot of these mediators increase in the serum, in the nasopharyngeal aspirate, and in the nasal absorption sample. So, there is some comparison across the different sites, and also over time. It's quite difficult to characterize time in these rather uncontrolled investigations when people have been infected at different moments, and their recollection of when their first symptoms appeared is hard. And I'll mention at the end how we can get over that by doing human infection challenge in volunteers. Okay, so what came out of that? This was, I think, one of our greatest accomplishments, and you'll see Anna Gara here. Is Anna in the audience? So Anna and I have worked together for many years, and this was a real collaboration between Anne and ourselves, looking at the transcriptomics over time. And you can see that there is a change in the levels of these mediators with severity, but also looking at the transcriptomic signature in the peripheral blood, which you can see in a pictogram here. The antiviral molecular score is only seen early during the infection and then declines to be replaced by this rather inflammatory marker, which in Go terms is characteristic of bacterial infection. But after a lot of ferreting around in the data, we're pretty sure that most of that signal is just due to influenza, and that the pathways involved in antibacterial defense are actually activated just by severe flu in the second week of the disease. So I think this was an early attempt to try and look at the time and the place at which inflammation can be monitored, and was very groundbreaking at the time, but also I think was groundbreaking in the way in which we managed to collaborate. Now, this is another timeline of Mosaic. This is probably one of the most well-known slides to come out of Mosaic. This was made by Maria Zambon, and it shows how difficult it is to get the timing right using conventional grant funding mechanisms when you've got a pandemic on your hands. So Wellcome invited me to put the consortium together soon after Easter. We got it in within 6 weeks, which I think, considering the size of the consortium, was amazing. The referees were quite quick, and then there was a lot of legal and financial wrangling over the summer. And we didn't get the award letter until the autumn, and then Imperial finally allowed us to advertise for staff and start working on the pandemic. And this was an expedited process, which was as fast as we could go at the time. So we didn't want to be caught out, and we set up this sleeping platform in case there might be a future pandemic. And this was really maintained by Kenny Bailey and Callum Semple. So they were key young investigators on the Mosaic study. Kenny was looking after the host genetics, and has done some brilliant work on host genetics of infectious disease. And Callum is a professor of outbreak medicine in Liverpool and is a pediatrician. So I think this combination of a young talent who was willing to spend time putting together this sleeping platform was really instrumental in allowing us to respond very rapidly to our current pandemic. And this consortium, the Zurich 4C consortium, with the three of us as co-leads, has been amazingly successful. This is actually a bit out of date. So well over 240,000 patients now being recruited. At one stage, we were recruiting 50% of all admissions to UK hospitals. So this is a much bigger scale than the Mosaic, but we couldn't have done it if we hadn't done Mosaic, and we hadn't refined all our protocols and got all the legal agreements with over 200 different hospitals in place in case there was another pandemic. So this has given us a huge biobank of materials and a huge resource in terms of all the patient details that were collected by NHS staff on a WHO protocol that Jake Dunning went over to Geneva to refine in case of future pandemics. So I won't show you all the outputs from this. It's been a huge amount that's come from it, not only in terms of academic publications, but also in terms of the advice that's been provided to government and to the NHS on an almost daily basis in terms of how things are progressing. But this is just one of the scientific publications. So this is the wonderful Ryan Thwaite, who I'm very pleased to say has now been appointed as a lecturer within the National Heart and Lung Institute, looking at the interactive network of different mediators, and you will see some mediators here to do with thrombosis and inflammation of vessels, but also IL-6, which we know to be an important therapeutic target. And interestingly, GM-CSF, which in the mosaic samples was not raised, but in fatal COVID is very significantly raised. So it's allowed us to make some distinctions and pick up some differences between these different types of infection. So ISRIC is an ongoing success. It's a very open platform. People can request samples on condition that they feed results back into the network. And it's a massive resource. I think no other country in the world has managed to produce a resource like this in terms of enabling science widely. So just wanted to mention the other big thing which has been going on, which is Human Infection Challenge. And we're just coming to the end of this wonderful consortium called HICVAC, which I lead, co-lead with Andy Pollard from Oxford. So this is Human Infection Challenge Consortium funded by the MRC and BBSRC to try and accelerate human challenge work. We've done a lot of work with influenza and RSV challenge, which have different time courses and characteristics. And we're now just beginning to report our first studies of SARS-CoV-2 challenge, where we instill the very smallest dose we can measure in the lab into the nose, and we get 50% infection rate with a beautifully reproducible set of symptoms that resolve over time, and with very clear viral kinetics, beginning in the throat, peaking in the nose, and then resolving. And if we work out how much we're getting in terms of virus coming out of the nose, you'd only need a globule of spit or snot, which is about 10 microns in diameter, in order to deliver the infectious dose that we used to challenge our volunteers. So I think, to sum up, it's all about timing and location, and the mucosa is absolutely vital. We have to develop methods of monitoring the mucosa, and this is a wonderful, exciting, 10-year plus collaboration that we've been doing with a whole team of people inside Imperial and outside Imperial, including Crick investigators. So thank you very much.
Thank you, Peter. Any questions?
Thanks. Just wanted to ask you clearly do your human challenges, which are fantastic, but there is one of the issues that clearly from our large, that human challenges you're doing, I assume, are in the health people in fear, so which can control the young.
Yes. Yeah. Yeah.
Well, clearly the problem in the real life is that in the people my age, whereas they are clearly going to be facing the challenge. So do you think that your picture is adequate to answer the real life question? Okay,
it is inevitably a problem with human challenge is that you're not choosing people who are highly vulnerable. That would be too risky. I mean, with the RSV challenge, we've actually gone into older adults, the 60 plus, going up to 75, and we've got some results. This is in collaboration, I must say, with GSK, supported by the MRC through a study called Inflammage, which is part of the Eminent Consortium. And we're finding in those older adults that we are getting enhanced disease. It is possible to do challenge studies cautiously in the more susceptible, but I wouldn't propose challenging infants with RSV. Infants are clearly a group that you would like to challenge, ideally, but we just can't do it. I think that what we get from the studies of hospitalized patients is the severe end with limitations. What we get from the challenge study is the mild end with limitations, but matching the two together, I think, gives us quite a nice picture.
So we have time for one last question.
Yes, sir. Thanks very much. As a concept of marina, it was very low.
Well, certainly. This is one of the aims of setting up the Human Infection Challenge, is to be able to rapidly test vaccines and other treatments, interventions, looking at the timing. And we know that the timing of steroids, the timing of antibody therapy is quite crucial. So again, it's place and time. And it does greatly accelerate the validation of new treatments and new vaccines. I must say that in the human challenge with SARS-CoV-2, we built in remdesivir as a rescue treatment initially, but we saw basically no effect, a very slight depression of viral load during a period of 5 days of treatment, but the viral load then immediately went back up again. Although it is an antiviral, it has very, very little effect in the model. Sorry.
So the next speaker is Professor Michael Onstein from the Department of Computing at Imperial College London. He will tell us about geometric deep learning for functional protein design.
Sorry, can I try to disconnect the... Okay. Thank you very much. So really great pleasure to be here. I think it's my first offline talk in probably almost 2 years now, so it feels a little bit strange. I think I got used to, probably like everybody else here, to doing talks over Zoom. So thank you very much for the invitation. I guess I'm a little bit different in my background from probably most of the people sitting here. So I'm a computer scientist. I'm professor at Imperial College in the Department of Computing, and where I hold the chair in machine learning and pattern recognition. And I would like to briefly describe our collaboration in the past several years with the group, the PFL, the Protein Design and Immunoengineering Lab, headed by Professor Bruno Correa. And as you probably know better than me, proteins are really interesting, well, because of their obviously, their fundamental importance in biology, but also obviously in a clinical setting also as important drug targets, and therefore it's crucial to understand how proteins work, how they interact with each other. And one of the crucial problems in bioinformatics was to predict the structure of the protein from the the amino acid sequence, the so-called protein folding problem. So it was conjectured that the sequence fully determines the protein structure already in the 70s. It has taken quite a while to get to accuracy that would probably be acceptable with recent breakthrough from DeepMind. We're actually interested in the inverse problem, so to say. So we're trying to design a protein that has a certain function or certain structure that endows it with this function. And here, I think a good metaphor would be this lock and key that was already proposed by Emil Fischer in the late 19th century. And here, of course, it's only a metaphor because in reality, the problem is more complicated. It's not only geometric structure that is complementary, like in lock and the key, but there is also, basically, these are charged objects, right? So there are some forces that repel or attract different parts of the molecule. And somehow the standard mindset in the protein world, in structural biology and bioinformatics, was that we have these one-dimensional objects, the sequences of amino acids, then they hopefully allow us to predict the structure. Then from the structure, we can go to the function. But of course, it's a little bit naive and wishful thinking picture because we can have examples of proteins with similar sequence, but dissimilar structure. You might have some unhappy mutation that completely breaks the structure. You might have dissimilar sequence that folds in a similar way. And there are many such examples as well. And finally, you can also have dissimilar sequence, dissimilar structure, but similar function. So completely different proteins bind, for example, the same target. So the way to even basically where to start when we try to think of protein function, of course, you can think of proteins in many different ways. So the different representations, either atomic point clouds, or you can think of them as maybe as graphs. The so-called stick diagrams, or maybe as secondary structures. So this is what is typically visualized in the form of ribbon diagrams. We believe that molecular surface is a good abstraction for certain types of problems involving proteins, because it allows us to focus only on the part of the protein and represent only the part that is interesting for interactions and abstract out all the internal intricacies of the fold. And I think this picture visualizes it nicely. So when these two proteins that are here presented as these plastic models interact, it doesn't really matter what happens inside. So the special kind of tools in machine learning that we are developing for these and similar kind of applications is what we call geometric deep learning. So it's in general a principled approach to machine learning problems that I don't have time to go into deeply today, but basically, you can think of it as how to apply deep learning architectures to geometrically structured data, such as surfaces and graphs. And in this case, we are modeling the outside molecular surface of the protein molecule. We're represented as a mesh on which we can construct a kind of local operation in the form of what we call geodesic patches, and apply something similar to convolutional neural networks that are very popular now in image analysis and computer vision to these different geometric objects. So basically, we decompose the molecular surface into these patches in which we compute geometric and chemical features. So the geometric features are computed from the surface itself, representing its curvature and other geometric properties. Chemical properties are related typically to the charges, to the basically electrostatic properties of the surface. And basically, this gives an architecture that is similar to convolutional neural networks that are popular in deep learning that can be also specialized for a particular task. And the kinds of tasks that we can consider, there are multiple, of course, so it's a generic architecture, but specifically we looked at problems of predicting interfaces, so the site on the protein surface where the protein might interact with another protein. We also looked at pocket classification, so predicting small molecule ligands, and also large-scale protein-to-protein interaction search. So we want to have, for example, a target protein, and we want to identify proteins or maybe some small peptides that would be able to interact with this target. So we are able to identify some seeds on top of which we can build protein binders. So you can think of this as one of the first stages in the design of future biological drugs. So let me show you briefly the interface site prediction task. So essentially it's a point-wise classification problem. So we are interested in labeling the probability at every point of the molecular surface, how probable it is to belong to an interface or not. So in this case, the training set comprises examples of interface and non-interface points. The criterion is area under the curve, the ROC curve, and we use multiple data sets of pairwise co-crystallized proteins that are known to be binders. So about 3,000 such protein pairs, 90% of which are used for training and 10% are used for testing. And this is the kind of architecture that we use. So we get very good results. So it's very accurately predicts the interfaces. And here you can see some examples. So this is a protein that was engineered basically to bind to some target. So this is the wild type that occurs in nature, and this is the modified, the engineered protein. So you can see that the wild type doesn't bind well, and the designed one does. And this is also very nicely reflected in the predicted interface score. So the ground truth here is shown in green, and it nicely overlaps. Here is another example. So this is also a comparison of wild type and design, and you can see that the interface again is highlighted very nicely. So the problem of very fast protein-to-protein interaction search. So in this case, we try to build a local descriptor that would be indicative of interaction, of binding. So this is what is called in machine learning a Siamese architecture. It is trained on triplets of points. So we have a pair of positive and a pair of negative points, basically interacting and non-interacting. We use a special loss function that we call the d prime loss. So it doesn't matter technical details. And we have about 6,000 interacting protein pairs, 80% of which are used for training. So here again, we can see a clear distinction between interacting and non-interacting patches. So we can nicely train this network. And here you can also see an ablation study that shows that the combination of geometric and chemical features produces the best results. So the area under ROC curve here is 99%. So it's extremely accurate. And the problem that we are trying to address is using again this lock and key metaphor is when we have multiple locks and multiple keys, we try to match them to each other very fast. And here is an example of a benchmark. So the standard way it is done is by looking at the number of resolved complexes that are ranked at a certain position. So you can see that here, well, we can outperform standard methods such as PageDoc, but also the speed at which this method works is about 4 orders of magnitude faster. So it really allows dealing with multiple proteins, potentially even entire protein-to-protein interaction networks. Let me skip this additional result. So an example of application, for example, well, talking about immunology, so the checkpoint immunotherapies are one of the probably interesting and exciting directions for therapies against cancer. So in this case, you can see an oncological target. So this is PD-L1 protein, where we can use this method to identify the binding site. Well, in this case, it is probably well known. And then we can search a large database of potential protein seeds that are likely to bind this site. And then we can put them on a protein scaffold. So the usual way that it is done in this field. And here you can already see a crystal structure for some of the protein binders for this target. So you can see, well, again, I'm not an expert in proteins, probably you can tell better, but here these are completely different protein structures that were designed that bind these sites. So you can see, for example, one helix that goes along the interface and 2 helices that go across. So basically these are de novo design proteins that never occurred in nature. Just one word about follow-up work. So we were also able to significantly accelerate. By significantly, I mean by up to 3 orders of magnitude, all the computation, make this framework totally differentiable. So we can now basically backpropagate and design the protein as part of this network. So this is thanks to a special library that my postdoc, Jean Fidi, has been developing called kernel operations, or KEOPS, for GPU. So I will finish here. I think it's an interesting approach, a novel tool set for protein science. It allows to do task-specific descriptors and representation for protein structure and functionality. In many applications, particularly those that we considered, it's significantly more accurate and faster than previous methods by orders of magnitude. It potentially allows to deal with undruggable flat interfaces, so protein-to-protein interactions are potentially interesting as drug targets. What is also important, and this is compared to methods such as AlphaFold, it doesn't at all rely on any evolutionary history. And this is important in de novo design because we are trying to design proteins that never existed in nature. So I will stop here. Thank you very much.
Thank you, Michael. I would ask people that have any questions to please use the microphone.
Hi, I'm just wondering about your design a protein-resistant nature. Are you concerned of the possibility that that will create an immune response? Because that is clearly the first thing that we would have.
Yeah, so that's an excellent question. So I'm not an expert, so take with a grain of salt my answer. So one of the possibility, and this is what my colleagues in Lausanne are doing, so they can take a human protein and modify it in a way that will bind better, but it is still basically something that the immune system is familiar with and probably will not create immunogenicity. But again, I think this is a big topic and a big problem of biological drugs in general. So I don't think we have really a silver bullet. So this is a computational method.
Any other question?
Yes.
Could you please use the microphone, thanks?
Sorry, fantastic work. So you generate these virtual therapeutics with high fidelity and quick speed is what I read from that. I mean, how engineerable are the solutions?
Yeah, good question. Yeah, so it takes, of course, some time to basically to do the experiments and to make sure that the design works. So I think out of the engineered binders, so we had something like 5 candidates. 1 failed to crystallize, so we were unable to get the crystal structure. 3 others got a crystal structure that is has distance smaller than 1 angstrom, so it was very accurate.
Thank you, Michael.
Thank you.
The next speaker is Mark Brinja from GSK. He will tell us about immunology research, leveraging novel immune genetic insights into new mechanisms to find future medicine.
Great. First thing to say is actually thank you to Peter and Charlie and many people here at the Crick who did some amazing work through the pandemic, and it's really appreciated. And also thanks to Steve and Guido for the huge energy they've put into building this network, and we're really excited to be here to talk about some of the areas that we're focused on in GSK. And just to highlight, actually, in the new GSK strategy, what we're feeling now is that immune dysfunction actually potentially contributes to the pathology of many more diseases than we'd previously appreciated. And the R&D focus now, and the whole strategy, is really to focus on the science of the immune system and to really start to apply human genetics and advanced technologies to think about new ways of interpreting those insights from immunology. So I'm leading the immunology research unit at GSK, and the way we think about how we target select for what are those molecules that we need to generate in designing new immune therapeutics, we're really focused on generating new insights from human genetics. And as we look at those, we know from previous understanding from great work in the field of how these potential surface receptors
the signaling components in the cell, and the transcriptional responses and the outputs of those potentially contribute to disease. And then we follow the science from understanding whether it's the T cells or the B cells, stromal cells or myeloid cells that impact that, to think about where is the best therapeutic opportunity. But what's clear is that genetics doesn't always tell you about which cell type, it doesn't tell you about which molecular pathway, and it doesn't tell you about often directionality or which are the best potential diseases to follow. So part of our strategy has now actually been to start to think about using deep tissue immunophenotyping, being able to access samples from patients, taken plus or minus treatment, and apply many technologies like RNA-seq, single cell sequencing, and that pathway analysis, and by also combining that with our ability, once we identify the pathogenic cell types, with genome-wide capabilities to apply functional genomics, to really triangulate from genetics, the deep tissue immunophenotyping and that functional genetics, to actually start to uncover novel insights into dysregulated cell types, which are the pathways that are potentially really driving disease that are differentiated from the mechanisms and molecules that have previously been evaluated. Try and understand at the molecular level those immune mechanisms and pathways, and from that, trying to get to new targets that are going to allow us to develop differentiated medicines. And through that, also start to understand what might be the future clinical opportunities for those. And while often we might start with human genetics of, say, rheumatoid arthritis or lupus, some of the insights that we might get from these might tell us actually the best place to go might be in neurology, might be in oncology, or it might be in nephrology. And so starting to be disease agnostic, to really follow the molecular insights and those mechanistic insights that come from understanding how those pathways are changed in patients and diseases, allow us to do one of the things that we're thinking about in GSK now, is to be disease agnostic, to be unconstrained by those areas where you originally started the research, to be able to follow the science, and to start to think, well, if we've identified a target that started out as, say, a lupus target, how do we really get into the right patients? And talking to Paul earlier, an area that we're increasingly interested in is, what are the mechanisms of immunoinflammation that potentially impact on central mechanisms of neurodegeneration? Those migratory cells at the barrier, whether that barrier is in the lung, the skin, the gut, the blood-brain barrier. How do those cells and that interface of the immune system with all of those other organs actually allow us to move forward and get new insights into diseases that previously hadn't been considered as being purely immune-mediated? And the thoughts there have been that the inflammation was essentially a response rather than a driver. And that ability to look at chronicity, that transition from acute inflammation to chronic inflammation, and what are the cell and pathways that potentially modulate those potential intervenable points for us. And one of the observations that we've made as we've followed the science from the genetics through functional genomics and deep immunophenotyping is that we also need to start to invest in and build data sets that allow us to go from those genetic associations. And we've built big data sets from our collaboration with 23andMe, our collaborations with FinGen and UK Biobank, and the GWAS catalogs, to actually then start to think about what are the causal SNPs, how do we do deep immunophenotyping to understand where the genes that are associated with those parents are expressed, which direction do those expression changes occur, and where are they normally expressed. And we're doing some work with the Altius Institute in Seattle to do some deep immunophenotyping in CD3 cells, monocytes, T cells, NK cells, Tregs, to really start to understand at a molecular level how that genetic variation is potentially affecting genes, and through that narrow down the cell types that we think are potentially driving those pathogenic processes. We've got a fantastic collaboration with open targets up at the Sanger Institute with the Wellcome and EBI. And we're building, obviously, through interactions with big consortia like the ANP, to build access to samples from RA and SLE patients. And we're just about to sign an agreement for the next round of ANP. And we're trying to get access, obviously, to really important data sets in IBD, in SLE and RA, but also strive to do much deeper phenotyping of those samples that we got from our own clinical studies to really add to these data sets so that we can then go from those cell types, really start to understand which of those potential targets have actually got the real opportunity to deliver differentiated function that aren't just giving you another anti-TNF or another CD20, but can really give us future medicines in those differentiated areas. As we've followed that scientific trajectory of going from genetics through disease understanding, we've actually found ourselves developing a portfolio, and these are just some of the diseases that we're now starting to work on based on that strategy. And we've got assets and molecules that are now targeting OA, atopic dermatitis, etc. And what we see is that strategy is taking us into new areas, new diseases that we don't have experience of, that we don't have access to patient samples. And one of the reasons we're particularly excited about the opportunity of thinking about this immunopathology consortium is how do we then get insights into things like sarcoidosis, androgenic alopecia, and those non-traditional immune-mediated diseases. And I think the ability for companies like GSK to work fantastic scientists like yourselves at Imperial and across London and the UK, I think is a really important opportunity that we're very keen to explore. So I will stop there. Just a very brief overview of the strategy, really focusing on genetics, giving us new insights into immune mechanisms, thinking about treating immune diseases in a different way, and thinking about different diseases in an immune way. And I think as we converge from those two angles, I think it's an amazing opportunity for us to work together. So happy to take any questions.
I think that's really good, and it's great to see a sort of original approach how to go about medicine discovery. What happens if you sort of road test some of the established medications and imagine how these would have gone through the pathway? Would they have been discovered using this type of approach?
No, great question. And one of the things that we are doing from the genetics and from the functional genomics aspects is actually looking at all of the licensed medicines to see how they behave in those cellular systems, essentially as a sense test or a road test, as you say, to validate that the phenotypes that we're measuring are actually consistent with being predictive of potential clinical opportunity. So that's exactly what we're doing. Thank you.
Hi, great. I just wonder exactly if for your approach you have noticed what probably is already well known, kind of very striking difference in the genetic contribution between different ethnic populations. So the kind of approach we do in clinic, which we give the same drug if you are white, Caucasian, or Black, African, Caribbean, is probably totally incorrect. And we do have some evidence for that, but I just wonder how much this is emerging from your approach.
Great question, Rena. I think one of the observations that you rightly make is not only in the way we clinically treat patients as being very biased by traditional setups and experience. Also in the genetics data sets, we're very, very clear that in UK Biobank, there's an extreme bias for white, middle-aged individuals. And one of the things that we're doing together with 23andMe and FinGen is trying to specifically recruit cohorts. And that's particularly relevant for things like lupus, where the prevalence and incidence of SLE in the black population is very different. And what we're seeing from the genetics is potentially some of the drivers might be a little bit different as well. And it's early days, but I think we're certainly very keen on increasing the diversity of the genetics data sets that we access, but also increasing the diversity of how we think about accessing translational science.
We have a question online from Matthew, that's my last name. Could you please comment on difficulties in developing consortia between NHS, university and industry, for example, in studying diseases such as lupus?
Thank you. I mean, I think I have a very positive experience with our AMP SLE data sets, where that's a big consortium with many pharma and many academic collaborators, which has worked very well. I think one of the challenges with setting up consortia and collaborations, particularly in the UK, is many of those clinical cohorts seem to be very fragmented. And actually the hardest part we find is getting academics to talk to each other and to come together in a collaborative way. We've made a lot of progress over the last few years, but it's certainly one of the challenges that we are working hard on.
So I just start by giving a little intro into atherosclerosis, which of course is the most common disease of arteries. I mean, arguably it's the most common chronic disease. It starts then with low density lipoprotein or LDL permeating into the vessel wall from the blood. And this leads to signals that bring in leukocytes attaching to the vessel wall. And macrophages get in there and start taking up the lipid, become engorged as foam cells, they die. There'll be some immunological activation. There's a reactive chronic inflammatory process bringing in a fibrous cap that is protecting that necrotic core from the blood. And then eventually the end game is that that cap will fracture or rupture, leading to the access of the clotting system to the highly thrombogenic necrotic core, and you end up with an intravascular thrombosis. But this series of cartoons was put together by Russell Ross some 25 years ago now. And Russell Ross, I think, is credited really with kick-starting the current view of atherosclerosis as an immunology-driven chronic inflammatory response to antigens deposited in the arterial wall, particularly chemically modified lipoproteins, or I should say, well, lipoproteins come in unmodified mostly, and then get modified in the vessel wall by reactive oxygen species. So the macrophage is in some ways center stage of all this, obviously being controlled in large part by upstream immunological drivers. But the physiological uptake of LDL by the LDL receptor is the way to dispose of the LDL in the vessel, and is actually a physiological process, and it's homeostatic, but it's controlled by receptor downregulation, so the macrophage can't take in too much lipid. But once modified, oxidatively or otherwise, the LDL can be taken up by scavenger receptors. This is not regulated and results in the foam cell formation. And these cells eventually die and release pro-inflammatory cytokines and growth factors, and exacerbating the chronic inflammatory process. So one of the main drivers to our interest in this process is the homeostatic principle that the humoral immune system actually has the potential to keep modified antigens, such as LDL, out of the clutches of macrophages. And this goes back to a program grant that Marina and I shared together some years ago, where we were looking at the protective role of the complement system, the upstream complement system, and IgM in forming complexes on modified particles and removing them for safe clearance. And I think this is probably the key experiment that Miles Lewis, a PhD student, did, showing that mice deficient, these are LDL receptor knockout mice, this is an atherosclerotic model, and these mice, if they're deficient in IgM or deficient in C1q, get actually very surprisingly exacerbated disease, particularly the IgM deficient mice, surprisingly accelerated atherosclerosis. So IgM
in this mouse model is protective. Well, can we take that into humans? Well, that's exactly what Ramzi Kamis has been doing. He's a cardiologist. He's actually running now an immunocardiology clinic. So he's keen to collaborate with immunologists. And he's done a number of studies now with large numbers of patients, big trials. And the bottom line is that high levels of total serum IgM, including IgM anti-MDA-LDL, are associated with protection from coronary adverse events in humans. Now, we want to get away from mice, and unfortunately, the block here is getting tissue. So if I was to say, can you give me some blood vessel, please, for my research, it's unlikely many of you would volunteer. So we're interested in developing sophisticated imaging techniques. Ramzi is an interventional cardiologist. He's done some very nice intracoronary imaging and established that low levels of IgM antibodies correlate with coronary plaque characteristics that reflect stability, such as small necrotic cause. And so this for the first time actually links IgM, low levels of IgM, to objective risk of plaque rupture. So the questions for the immunologists here are, what are the underlying mechanisms regulating protective IgM levels, and can they be therapeutically controlled? So going on then along this imaging line, there is potential obviously for molecular imaging of atherosclerosis, and in the lab we've made two in-house generated monoclonal antibodies. The first is called LO1. It reacts with MDA conjugated LDL, and on the right you see an example of near-infrared fluorescence imaging with the tomographic machine that we have on the Hammersmith campus, with a humanized Fab fragment of LO1. It was originally a mouse monoclonal autoantibody, and in a collaboration with GSK, this has been humanized and rendered into Fab and single chain versions for clinical use. And the LO9 antibody is actually particularly interesting, as it's unique of its kind. It reacts with native LDL as long as the LDL is glued down to a surface. So it reacts with a adhesion-dependent allosteric epitope in native LDL. And Cinzia Marchedu did a PhD in collaboration with Jan Pascio at In Vitro, in which she showed that this antibody could be used for pre-targeting atherosclerosis. So basically, the antibody is conjugated to trans-cyclooctene and then injected and circulated for 72 hours. And then once the blood pool has cleared, fluorine-18 tetrazine is injected. And what you can see here is the foci of early atherosclerosis. This is not modified LDL. This is by and large native LDL that has adhered down into the tissues. And this distribution is completely different from the distribution of oxidized LDL. So it gives us two different approaches to looking at vessels in humans, potentially. So I want to shift now to the work of Joe Boyle. So going back to the macrophage, just like to start by just saying a little about angiogenesis. So as the plaque develops, it becomes hypoxic and brings in new blood vessels. The big problem is that these new blood vessels are very poorly developed and tend to fracture and rupture, and they bleed. And intraplaque hemorrhage is a particularly pernicious thing to happen, because what it's doing is potentially physically disrupting the plaque and can lead to an acute event. But if it doesn't do that, it basically dumps erythrocytes into the vessel wall, which of course are composed of lipid, and they contain hemoglobin and iron. And these are all potentially pro-inflammatory. Joe made the extraordinarily interesting observation that he's a clinical pathologist, and he looked in great detail at macrophages in human arterial specimens, and showed that the macrophages, the lipid-laden macrophages, the foam cells, occupied a different zone in the tissues to hemosiderin-laden macrophages. These are the ones that are digesting iron. And in fact, the CD163 being a marker of what he now calls M-heme, these are macrophages that are dealing with blood, tend to be HLA-DR negative, and the HLA-DR negative tends to be CD163 negative. So he's done a lot of work on this. He's spent 10 years studying this, and with transcriptomics, he's shown that this is a different profile transcriptomically to classic M1 and M2 macrophages. And you'll have to read his papers. I can't go into the detail, but he's worked out the whole signaling pathways, which result in these separation of macrophage ways. And just to end then, one of the points of the network is to create centers within it. And we now have a vasculitis center of excellence. This is run by Charles Pusey and Justin Mason. Justin is interested in, has a very large collection of giant cell arteritis and tachyarteritis arteritis, in which the events are quite similar in some ways to atherosclerosis, but they're taking place in the adventitia and the media rather than the intima. Time prevents me going on about that, but I'm sure he'd be interested to hear from anybody if you have, think you might be able to contribute. And then lastly, I'm showing you this slide of Makiv Kwajo and Mike Emerson, who are working on a complication of treating HIV with antiretroviral virus drugs, because these are thought to now be implicated in accelerated arterial disease. And they're actually working mainly on platelets and endothelial cells, but I think they have room to start studying T cells and macrophages. Thank you very much.
Time for very quick questions.
Thanks, Doria. I just want back essentially to a science. We know that in lupus atherosclerosis is accelerated, but we also know that rituximab B-cell depletion drives IgM that stays low irrespectively of returning of the B cells. So I wonder your thought about it, because it's been intriguing for a while. All my patients are practically IgM depleted almost.
Well, many years ago when rituximab first came of use in rheumatology, I remember going to give a seminar at the company that makes it, and told them the news about low IgM levels. It wasn't of great interest to them. And I don't think anyone really particularly picked up on it, but I think it is very much something one needs to look at seriously. There is absolutely no question in my mind that low IgM is detrimental to blood vessels. Yeah, thanks for making that point. Hi, Jimmy.
Thanks, Dorian. Is it total IgM that seems to be important? And I guess if so, there are now, I think, huge studies of genetics of total IgM, etc., UK Biobank, but others as well. And I wondered if that might help address the question of correlation, causation, and you could look at are genetic variants that predispose to higher or lower IgM protective against coronary artery disease?
Well, that's absolutely a terrific contribution. I will talk to you about that. It is total IgM, as well as if you look specifically at antioxidase LDL antibodies, but they're both protected. So it actually, in the mouse, the innate immune response, the IgM response, is a large part of it is devoted against oxide lipids. If you do an experiment where you do a sequential hybridoma experiment and you clone out and test the hybridomas, up to 50% react with MDA-LDL from a atherosclerotic mouse. So this is a common determinant. We'd love to talk to you about that. We'll be in touch. Great idea. Okay.
Great, thanks. So we're having a bit of a swap round. So I'm pleased to welcome Alan Melcher from the Institute of Cancer Research from Imperial. He's going to take the next talk.
Sorry, I might just need a bit of help in switching to my slides. That's,
Thank you.
you didn't either on that person. So.
You too.
Shut that one.
Is it loading?
It's on the desktop.
Okay.
Oh, there we go. How many? Getting closer. Oh, there we go.
You can just get escape, yeah.
I'll try. Start.
You find it?
Yeah, look this way, switch.
That's that one. I need to. Oh.
Done. You okay?
I think so. Don't go anywhere. You'll see it. Oh, something's happened.
Is that the
That looks like the end. Yeah.
Okay.
How do you get to the beginning? Better share. That's good.
Maths are so much easier.
They are, aren't they? There we go.
Okay.
Exactly.
Okay.
Sorry about that. So thank you very much, and thanks again for the invitation, particularly for including ICR in this network. It's very exciting for us to be here as a relatively small institute, I guess, in the context of some of the others. But I hope I can show to you and talk a bit today about what we're up to and where we fit in terms of the cancer. Obviously, we're entirely cancer focused, and where particularly within the clinical, as an oncologist myself, the clinical context of immunotherapy and some of the challenges we currently face. So the first point is very briefly that actually cancer immunotherapy has been around a long time. This very ancient slide shows that actually even to single dose, high dose interleukin-2, you do get some response. This is a progression-free survival curve in patients with advanced melanoma. And basically what you see is that some people do survive even with this crude therapy. Other therapies, particularly, for example, giving BCG into the bladder for non-muscle invasive bladder cancer, works very well. And don't forget, vaccinating against HPV is a major anti-cancer immunological intervention now. But of course, the major multi-million-dollar drugs now are these checkpoint inhibitors. And this is the start of the checkpoint blockade revolution, and I think it's fair to call it that. This is now, it's only 2010, so we're only talking a decade ago with ipilimumab, an anti-CTLA-4 antibody. The details don't matter other than a couple of points on here. One is, again, you see this tail on the curve. So unlike with chemotherapy or targeted small drugs for advanced cancer, we always use the C word with caution, but we are now curing patients with metastatic melanoma with 4 injections of an anti-CTLA-4 antibody now given up to 15 years ago. But the response rates, of course, were very poor. In the checkpoint blockade world, things have got much better. And now, at best, combining an anti-PD-1 with an anti-CTLA-4, we're up at around the 50% long-term control rate. And note that this is all, very exciting. And my slides have inevitably gone a bit pear-shaped. But bear with me. I think I'll just plow on regardless, the joys of Mac. What I should say then, so these drugs are particularly targeting negative checkpoints in the cancer immunity cycle, which I'll come back to. So what do oncologists always do? What they will do is add their new treatment, in this case, immunotherapy, to their current standard of cares. And you can't see properly on this slide, but suffice to say, in non-small cell lung cancer, if you add immunotherapy with an anti-PD-1 to chemotherapy, which was standard of care, you improve outcome for patients. Similarly, in radiotherapy, and I'm a clinical oncologist, a particular interest of mine, here's a trial comparing standard treatment, which is a combination of chemotherapy and radiotherapy for non-small cell lung cancer again, with and without addition of an anti-PD-L1 antibody in this case, again with significant improvements in outcome. So that's all very well and very exciting, but those drugs which are targeting those couple of negative checkpoints really remain the mainstay of immunotherapy in the clinic. And this cancer immunity cycle on the left-hand side of your screen just highlights all the positive immune checkpoints in green, the negative checkpoints in red, and makes the point that actually we're still very restricted in the drugs that have come through to widespread clinical application. And there have been quite a lot of failures. So on the right is a trial of anti-PD-1 plus or minus an IDO inhibitor, which looks very promising in preclinical mouse models and even in early phase clinical trials, that was remarkably negative when it came to the big randomized phase 3s. So where we are at the moment in the clinic is essentially everything is having to build on the current standard of care, which in immunotherapy really is about the PD-1, PD-L1 axis. Anti-CTLA-4 hasn't got much further other than in melanoma. And what it means, and I think the take home that I really want to get across, is we do not understand how cancer immunotherapy works. And I always used to talk about everything we did in the lab was lagging at least 5 years behind the clinic. I think that has now switched in the world of cancer immunotherapy. The clinic and is charging ahead. Here we have a slide from last year with the various combinations, 1,300 new combination trials, 222 targets opening over 2 years, but we really don't understand, quite frankly, what on earth we're doing. And our view is I think we need a much better integrated basic to translational to applied approach to understand. And that's what we're trying to do at the ICR. So when I moved down about 5 years ago, I was told, oh, nobody did cancer immunology, they weren't interested. But actually, everybody does it. If you think about it, all treatments are immunomodulatory. I've already touched on the chemotherapy, the radiotherapy that we do. Targeted drugs are also modify your immune system, so does surgery. So everybody actually has been extremely engaged and interested, but coming at immunology from a non-immune perspective, if that makes sense. So we have a major cell biology interest groups in DNA damage. And we all know that DNA damage can throw up neoantigens and can also mutational burdens and trigger CGAS sting and various sensing pathways. Also coming in from particular sites of tumor site interest are very large. We have a big breast cancer interest. They want to do and are doing a lot of immunology as well. I think there are particular opportunities in those diseases which currently don't respond to immunotherapy. Again, we don't quite know why. It's not all about the tumor mutational burden, but things like ovarian cancer, and you'll hear from Ian McNeish a bit later, sarcoma is another example. So to try and join up our working, not only across the ICR, but also with the Royal Marsden Hospital, we've set up this Centre for Translational Immunotherapy to try and coordinate the cancer activity that's going on in this space. We have the various divisions, as you can see there, and across the ICR centres, these are cross-cutting ways of working. We have convergent science, which is a big activity with Imperial, but also those are the various other areas that you can see, all increasingly linked to immunology. We're particularly interested not only in clinical trials, and the Marsden does many, but also in radiotherapy as immunotherapy. And the take-home from a lot of this is we have a lot of patient samples, including from standard of care treatments and trials which were never thought of to be due to immunotherapy, which may be of interest to address in the collaborative setting, and we're very keen to do that. So just as one example, briefly to finish off, here is a study on the left, which was led by Anna Wilkins, who's now here at the Crick, which was looking at preoperative radiotherapy for rectal cancer. So for rectal cancer, you'll have radiotherapy and then an operation. You can take a biopsy before radiotherapy, you get the surgical resection specimen afterwards, and you can see what's happened immunologically, and you can link that to the pathological response to the treatment. And on the left, the take-home is that in patients who respond well to immunotherapy, the good responders, the tumor changes essentially from non-inflamed to inflamed before versus after the radiotherapy. In other words, we talk a lot about tumors going from cold to hot. So that cold to hot change is related to a good response. You don't see such a clear link at all with the poor responders. And what we're currently doing is going back and looking to get samples from a trial called CR07, the clinical outcome of which was published over 10 years ago and showed essentially what it was about was that this preoperative radiotherapy improved local control of the cancer. But those blocks and slides are essentially still sitting there. So this now gives us much greater patient numbers of patients. We have about 80 or so given preoperative radiotherapy. And the Anna study was very much based around RNA transcriptomics, nanostring, not good enough for RNA-seq, but there's an awful lot you can do even with this very old suboptimal tissue. And we're looking to develop H&E, AI algorithms to characterize the immune landscape, etc. So the thinking is not only at all the samples we're collecting at the moment, but think about things that could even be done with a different view on things that were done quite a while ago. So to finish, there's no question immunotherapy is the greatest advance in cancer treatment in the last 20 years, he says, with no bias at all attached. But we really don't understand how it works. And we need to link up much better between mice, which have their place, so their limitations. We're very interested in human preclinical systems, sample analysis, but also now integrated into the design of clinical trials, in-depth translation, repeat biopsies on treatment, etc. Currently, we're really pretty stuck on the PD-1, PD-L1 axis, but as the technology advances, we're not, we can now look at old samples in a very different way, not just at the transcriptome, but what about the mutational burden? Are those epitopes that are thrown up by such mutations potentially immunogenic? And so it goes on. And I think where we would like to fit in and hopefully contribute, particularly to this network, is the collaborative opportunities arranged around our extensive trial systems. And these can either be academic, but they're also commercially sponsored. And we have engagement from big pharma to increase the translation. I think if there's any good side to the reality that, in my view, immunotherapy is a little bit stalled in terms of targets, it means that big pharma want to understand better as time goes by why their trials don't work, which quite frankly is every bit as important as why they do. And again, final comment, as has been mentioned earlier, the first workshop that we're setting up, and that's jointly with Jessica from Imperial, is in March of next year, and we're very much looking forward to that. So thank you very much.
Fantastic. Questions?
Sorry. Nobody's interested in cancer.
We're all very interested in cancer. Marina's
Oh, she's coming. Oh, she's coming.
I think you pointed out clearly the value of the human specimen. So I'm going to challenge you a bit. Right. What about the mouse work, which clearly has made the foundation of all this and is under a lot of scrutiny at the moment? So I worked in mice for many years, and I still want to work in mice for many more. But the question is it a valid criticism, or is just it's time that we change?
Well, I mean, I also have worked with mice all my life and continue to. So I think they have a very important role. But I think what is evolving, and of course it's not black and white, it's shades of grey, is that you can generate hypotheses in mice. We all know if you see the tumor in the flank of a mouse, it's not representative of the real thing. My personal view is humanized mouse models are a waste of, I'll rephrase that, okay, are limited in their benefit, because at the end of the day, a mouse is a mouse is a mouse. So I think you need to put together that mouse work, which remains critical, with the sort of particularly preclinical human assays, which we work a lot on, into those translational spaces as well, and go round and round that cycle. So if you generate data, and we've done this, and we do this, for example, with our oncolytic viruses, which is our major interest in immunotherapy treatment, we were able to show benefit in a mouse model, take that into a window of opportunity clinical trial, where we showed that an intravenous virus could get to a tumor and turn it from cold to hot. So it's the back and forth. We're not going to get rid of mice anytime soon.
I was very interested in the radiotherapy data, where you go cold to hot, and whether it's the radiotherapy that leads to survival, or whether your ability to survive the stress of radiotherapy tells you something about you and that you're going to survive.
Yeah, I think that's absolutely right. I think it's a correlation, and it is no more than that. And it throws up the hypothesis, which again, we do and can follow up in mouse models, that when radiotherapy turns the tumor cold to hot, but actually that's a very crude description, the heat of it. It's about the right heat at the right time and of the right sort. So it's absolutely just a correlation. I think it's a perfect example of where you then need to go back to the mouse, which is what we are doing, and trying to understand, and I won't, without going into too much, the doses, the fractionations, the areas that you treat. For example, in the clinic, we often treat the lymph nodes, the draining lymph nodes, because we worry there are cancer cells there. There's actually mouse evidence that that's not a good idea for immunological reasons. So it's going to be multifactorial, I think.
Thank you very much. Right, we're going next to Ed Tate from the Department of Chemistry at Imperial to talk about chemical immunology.
Great, well, yeah, thank you very much for the invitation to say a few words about what we do in the Department of Chemistry. So the Department of Chemistry at Imperial recently moved to the White City campus into a brand new molecular sciences research hub building, which puts us sort of right in the heart of Hammersmith campus. Also is out there, and a lot of small and medium enterprises, so a lot of companies as well in that area. There's about 70 PIs in the department, and about 40 of those work in this theme, which I lead, called the Chemical Biology and Healthcare theme. So it's probably among the largest chemical biology groupings in the UK, and quite a few of our PIs are also involved in the Crick Institute. So we have Louise Walpole and Ben Schumann, both of whom have quite large groups at the Crick. I have a satellite group, and Marco D'Antonio also.
Some of the areas we work in, and obviously we were kind of a technology-focused department, so we're looking for opportunities to apply our technology in collaboration with clinicians and with biologists. So we work on chemical probe design, discovery and synthesis. That includes things like small molecule chemistry, but also in the field, in a kind of emerging field of protein degradation, which many of you will have heard of, PROTACs, for example. Also in the area of design proteins, I'll say a few words about that today, and in nucleic acids, lipids, glycans, quite a wide range of different chemistry, let's say, that we apply to biological systems. We also do a bit of work in translational drug discovery, so particularly drug target identification and validation, so drug mode of action studies, for example, and also we work on drug candidates, one of which has been licensed into clinical trials and also into a spin-out, for example. We work on the chemical biology and analysis of dynamic protein and nucleic acid modifications, so post-translational modifications, for example, and epigenetics. And we work on new technology development, so things like molecular modeling and enzymology, and also nanoscale analysis and imaging. I'd also like to say a few words about the Centre for Drug Discovery Science. So this is a relatively new centre at Imperial, cross-faculty, aimed at maximising opportunities for internal and external collaboration in the drug discovery space, including target validation, also technology development. It really should give us access to a sort of nucleated set of technologies and resources across Imperial. The directors are Matt Futer, me, and also Mish from Cancer. We have over 40 PIs involved in this centre, and we have quite extensive biotech and pharma interactions, so please get in touch if you're interested in collaborating in this space. There are quite a few mechanisms for pop-up collaborations, I would say, so not least the Imperial Confidence in Concept scheme. So we do a lot of collaborations in that scheme, which allow us to sort of start collaborations every year, pretty much, in doing translational work towards developing our work towards the clinic. And we also have the MRS in Drug Discovery course, which had about 30 to 40 students every year. These students work on one project for a whole year, and many of them have also come to the Crick to do part of their project here. So that's an excellent mechanism to start new collaborations. These students are working pretty much exclusively in collaboration between groups between, for example, chemistry and medicine, or chemistry and biology. So just a few words about the kinds of technologies we developed. I mean, the first example I'd like to give is from my lab, where we developed monoclonal antibody PROTAC conjugates. So this is a technology, PROTACs are a technology which enable targeted degradation of proteins in cells. Usually using small molecules. So typically these molecules have been developed to degrade a specific protein with something that looks quite a lot like a normal small molecule drug. But they have many challenges, not least, well, they don't provide tissue specificity, and they also have some issues with uptake and pharmacokinetics. This is one way to enhance and overcome some of those challenges. Obviously, monoclonal antibody therapy is a well-established modality, but here what we've done is we've attached a PROTAC to a monoclonal antibody. And of course, this will give us the possibility to undertake targeted protein degradation in a specific tissue in response to expression of a particular antigen. So in an antigen-negative cell, of course, our PROTAC isn't going to enter the cell, and you won't see any protein degradation. But in an antigen-presenting cell, that antibody will be internalized, the PROTAC will be released, and you'll get targeted degradation just in that particular tissue type. This is a very powerful modality. It's catalytic. You have to deliver a very small amount of this molecule to a cell to enable rapid and also extended degradation of that protein target. The example that I'm going to show you today is a very simple one, using trastuzumab as the monoclonal antibody linked to a ligand for a protein called BRD4. It doesn't matter too much. This ligand could be any ligand to any protein of interest, via a linker to an E3 ligase ligand. And it's this molecule here that recruits an E3 ligase to induce degradation of a protein inside the cell. So it induces the formation of a complex between an E3 ligase and your protein of interest that wouldn't normally occur inside cells. So it induces the formation of that complex and degrades the target. This also features a linker, a hydrolyzable linker to the ADC. So this will be, once the molecule is internalized, it will be that linkage is hydrolyzed, it releases the PROTAC, and the PROTAC becomes active and can degrade the protein. This is a stable conjugate, in fact. And in a HER2 negative background, what we see here is, you can see a control lane here, which is a PROTAC, just a small molecule PROTAC that rapidly degrades the target. In the context of this antibody PROTAC conjugate, without the presence of an antigen, the BRD4 protein is maintained. You don't see any change in the amount of protein that's present. But when we incubate that with our antibody PROTAC conjugate, you can see with relatively low concentrations of this conjugate, we see rapid degradation of that protein target. So you could, in principle, put any ligand to any protein that you care about and you can put it in link to any monoclonal antibody you're interested in, and in theory target protein degradation to a specific antigen, also in vivo. I'd like to say a few words about the work of Nazila Kamali in our labs, in our department rather. So she's working on inflammation resolving nanomedicines. She's been working on this relationship between pro-resolving mediators and pro-inflammatory mediators, and making inflammation resolving nanoparticles, which can effectively deliver some of these pro-resolving agents directly into, for example, atherosclerotic plaques. That's what's shown here. So she's a specialist in formulating these drugs into deliverable vehicles and packaging them up with various different molecules in combination. And she's very interested in collaborating with anybody who's interested in delivering these kinds of molecules in vivo. She also does quite a lot of work with nucleic acid delivery as well. Francesco Aprile, who's in fact the representative for our department in this particular network, in the Immuno-Path network, is working on designer antibodies. So he designs antibodies from scratch to recognize specific post-translational modifications, for example. And he's developed these for purposes such as imaging and biological studies, of course, and also towards the clinic. So he'd be very interested in hearing from anybody who's got a particularly challenging antigen that he could potentially work with you on to develop an antibody against. Ben Schumann, who's got also his lab here at the Crick, has been working on precision tools to profile glycoproteins. So obviously glycans clearly very important in immunology. He's been developing metabolic labeling technologies that allow you to very precisely introduce what we call a clickable tag or a bioorthogonal ligation handle onto a specific sugar in a particular type of carbohydrate. So this will be incorporated into all of the glycans which are expressed on the surface of the cell, for example, with that particular sugar present in a glycan. He can then use bioorthogonal ligation chemistry or click chemistry to introduce a biotin or fluorophore, and that allows him to very selectively image or also identify the proteins that may be attached to that particular glycan on the surface of the cell. So very powerful technology for unpicking the glycan code, if you like. He also works on chemoenzymatic glycan synthesis, so the generation of novel glycans, which he puts into arrays with the group of Yan Liu in medicine. And for that, they've made microarrays of glycans, which allow you to screen for glycan binding. The group of James Waltonini and Marina Kromova have been working on carbon monoxide sensing. So carbon monoxide is produced endogenously by heme oxygenases, and it's immunosuppressive. Indeed, high HO2 expression has an impact on cancer chemotherapy outcomes. And they developed some very selective and sensitive sensors for CO levels inside cells. And they've used this to detect the CO that's produced in response to inflammation in a mouse model. And they can also simultaneously measure carbon monoxide and changes in cellular viscosity, for example, due to induction of cell stress or apoptosis, for example. So they're very interested in working with anyone who's interested in the immunosuppressive effects of carbon monoxide. Their molecules are obviously very easy to use. They're just small molecules that you can introduce directly into any cell in culture or even into an organism and immediately start measuring the CO levels as they change in response to different stimuli. Lastly, I'd like to say a few words about the work of Matt Fuchter. So he works particularly in drug discovery science and medicinal chemistry. So he works in small molecule medchem. He has many years of experience in this space. He's one of the inventors of a CDK7 inhibitor that's currently in clinical trials. And he also works on lysine methyltransferase inhibitors. For example, this dual inhibitor here, which stimulates an anti-tumor immune response in ovarian high-grade serous carcinoma. He's interested in drug delivery also, so if you have any interesting challenges in that space, I'm sure he'd be interested to hear from you. But he also works on photopharmacology, where we use light to switch drugs on and off, so we can uncage drugs spontaneously in cells and make them respond to light signals, giving us exquisite spatiotemporal control over the function of those molecules. So if you're interested in gaining that kind of control over any pharmacological agent, you should definitely go and talk to Matt. Lastly, he's also established a spin-out company with Hugh Brady at Imperial called NKIO. And this is based on fundamental discoveries in natural killer cell development and the pathways which modulate proliferation of these cells. So he uses some proprietary methods. I mean, these are small molecule therapies or small molecules which influence progenitor differentiation and expansion. And by this method, they can greatly improve the generation of these cells and also their tumor killing impact. And in a sense, this is small molecule cell therapy. So you're generating a cell therapy through the action of a small molecule drug. And this company is currently sort of in the early stages of development, it's received quite a bit of investment. Okay, I'd like to close there, and thank you all for your attention. Thank you.
Fantastic, thank you. Just about to stand up. Questions for Ed?
So for your ability to add PROTACs to antibodies, what's the sort of dosing level you can attach without losing solubility and selectivity for your antibodies?
Yeah, so that was a drug-antibody ratio. So there's this issue that if you add too much drug to an antibody, you can have solubility issues. I mean, I guess there are recent examples where that may have been slightly overturned, right? But yeah, we had a drug-antibody ratio of 4 for this particular case because we rebridged the disulfide bonds that you find in monoclonal antibodies with something that directly carries the drug. So you're matching the number of drugs attached to the number of disulfide bonds. But I think, yeah, they're quite big and sometimes hydrophobic, these PROTACs. So there may be a limit there. I think we have yet to hit that limit, but yeah. I mean, the benefit is having a catalytic drug, right, where you don't need to deliver very much of these compounds to the cell. Because you're exploiting the catalysis of the ligase. Any one molecule can degrade many proteins of interest by engaging multiple rounds of ligase activity on the same proteins of interest.
Thank you very much,
Thank you.
Ed. So our next speaker is Dr. Tatiana Ort, who from AstraZeneca, and she's actually online. So switch over.
All right.
She's going to talk about immunology at AstraZeneca.
Good afternoon, everybody. I hope the technology will work. And it says I cannot share the screen. Luis, will you be able to help? I'm sorry, it says you cannot share the screen while the other participant is sharing. Would it be possible to stop sharing? All right.
Yeah.
Yep, should work. Please let me know if you see the slides.
Your slide, just put it onto presentation mode. That'd be great.
All right.
Fantastic. I'll kind of interrupt you after 10 minutes as we're trying to keep on time, but take it away. Thank you.
All right. So just want to take a moment to thank Steve, Luis, and the organizer of the meeting for the invitation. Very happy to talk about the emerging immunology team and AstraZeneca. Was asked specifically to touch based on the Excuse me, is it a comment? Was asked to touch based on areas for potential collaboration, and I will do that throughout the presentation. My apology for not being able to join in person. I hope next year will be different. Again, my name is Tatiana Ort, immunologist by background, joined AstraZeneca about a year ago and heading immunology bioscience in respiratory and immunology TA. The next slide is a disclaimer, and we can move to the next slide.
Sorry, we can see the slides, but we can't actually hear you, so we're just trying to check.
Oh. So you cannot hear me?
Oh, we can now. Just need to go back to your slides.
Just need to go back to slides. Okay.
Yeah, now we can hear you, but we can't see the slides.
All right, let's try again. Share.
Perfect.
It's never worked like we want, right? So let me know when you see the full screen.
Okay, that's great. Yeah, thank you.
All right, let's start again. I want to take a moment to thank Steve, Louise, and organizer of the meeting for the invitation. Very happy to talk about emerging immunology team in AstraZeneca. Was asked specifically to touch base on areas for potential collaboration, which I will do throughout the presentation. Again, my apology for not being able to join in person. Hopefully next year will be different. Again, very briefly, my name is Tatiana Ort. I'm immunologist by training. Joined us at around a year ago, heading immunology bioscience in respiratory and immunology therapeutic area. So the next slide is a disclaimer. And if I move to the next one, just want to touch base and say a couple of words about our therapeutic area, respiratory and immunology. The goal is to develop transformative therapeutics, and we are accountable, our team, for both preclinical and early clinical portfolio. And the focus area in respiratory are asthma, COPD, IPF, and cough. And in immunology, we are focusing on core indications, which are diseases like lupus, inflammatory bowel disease, atopic dermatitis, and other within rheumatology, gastroenterology, and dermatology. The strategic focus, of course, novel and differentiated targets, innovative therapeutic modalities, patient segmentation and precision medicine, and transformative clinical endpoints. So let's talk a little bit more about immunology, and I don't need to convince this audience that there is a huge unmet need in immune-mediated diseases. Only 2 out of 29 million patients who is living with moderate to severe diseases are in clinical remission. And the gap between patient in need and patient in remission clearly highlights unmet needs, but also significant headroom for novel therapies. And there is a successful story in immunology, or one can say that psoriasis is one of them. Clearly, we are near cure in psoriasis with biologics like IL-23, P19, or IL-17 blockers. However, I think many will agree that psoriasis is one of the most homogeneous immune-mediated diseases, and we observe very different picture with disease like lupus, with disease like IBD, and with diseases like RA and AD, where some people refer that many options, treatment options, are reaching what it's called efficacy ceiling. If we consider why we are not achieving remission in majority of immune indication, and just very briefly, if we consider patient journey today, right? So on average, immune-mediated diseases have this relapsing remitting pattern. And after patient diagnosis, it's usually prescribed first-line therapies. If patients are not responding or losing response, they progress to second-line more advanced therapy. And loss of response typically means flares, and that is on a background of disease, which is progressive within the tissue damage and tissue dysfunction as well. So again, like couple of, it's of course $1 million question, one, we cannot achieve remission, just a couple of considerations, right? Firstly, we are dealing with very heterogeneous diseases, yet we're trying to treat everybody with the same therapy. Precision medicine might deliver therapy to the right patients and deliver it quicker, cutting time of cycling between the therapies. And this is, again, one of the areas which we are very interested in AstraZeneca. And we do have therapeutic options, although the therapeutic options which we have very narrow mechanism of action, right? Majority of them are cytokines blockers. So none of them directly address tissue pathology and none restore immune system to homeostasis. And there is also no treatment options for refractory population, which is quite big in some indications specifically. So we are much less aggressive compared to oncology in combination therapy, and we're much less aggressive in new modalities. Majority of immune-mediated therapeutics are biologics with very few oral options. So one of the pathway of interest for AstraZeneca Immunology is interferon, type 1 interferon pathway. As we all know, type 1 interferon are a group of molecules with pretty pleiotropic effects, which are a crucial link between innate and adaptive immunity response. Apart from their importance in antiviral immunity, type 1 interferon are increasingly recognized as a key player in autoimmune connective tissue diseases, such as lupus. It's known that type 1 interferon is present in elevated level in circulation in many patients with systemic lupus disease and contributes to pathogenesis. Anifrolumab is a human monoclonal antibody to type 1 interferon receptor, which will block majority of the action of type 1 interference. And it is developed by AstraZeneca and evaluated for the treatment of moderate to severe lupus. And on the next slide, this slide really illustrates the primary and select secondary endpoints from 2 phase 3 studies, TULIP-2 and TULIP-1, and 1 phase 2 study, MUSE, study for anifrolumab in patients with moderate to severe systemic lupus. And a dark blue checkmark circle indicates the statistical significant result. A light blue checkmark circle indicates that there was a numerical improvement when compared to placebo, but results are descriptive only, and the NS gray circle indicates non-significant result. Based on this data, and in this trial, anifrolumab was recently approved in USA and Japan for the use in patients in moderate to severe systemic lupus. And building on anifrolumab story, we are developing a portfolio of next generation therapeutics. And in the next couple of slides, I will describe our strategic priority and the areas for potential collaboration. First, about key modalities that we plan to apply for designing our next wave programs. I mentioned before, biologics already transform immunology markets. However, again, majority of existing therapy are single cytokine blockers, and they are reaching this efficacy ceiling in majority of immune indication, probably with exceptions of psoriasis. To break the efficacy ceiling and tackle this multi-pathway etiology, we target beyond cytokine inhibition and capitalize on bispecific modalities. With predominance of marketed biologics, immunology is poised for novel orals that might provide access to broader than biologics population. And orals is one of the key focus for our emerging portfolio. In line with rationale for bispecific, I would like to target signaling hubs and checkpoints upstream of multiple disease pathways and prioritize targets with shared cross indications to deliver franchise opportunity. And we are interested in innovative platforms and taking forward T-cell therapy program targeting immune tolerance. This is quite novel approach for immunology, where we plan to fully leverage capabilities and experience from our oncology colleagues with cell therapy CAR-T programs. And we all know that CAR-T programs is already in clinics in oncology and show some curative effect in hard to treat population. Second, we are committed to build insight from patient samples. And we are growing data sets from both our own clinical studies and external collaborations. So we are looking for opportunities to expand our translational cohorts and lupus, rheumatoid arthritis, IBD, and atopic derm. And this is one of the areas which we would like also to highlight as collaboration. Thirdly, what we're going to do with patients' data sets, and the idea here, and we definitely are interested in collaborating on novel science. We want to back translate knowledge from patients' data sets to precision medicine hypothesis, combination therapy, and of course, novel therapeutic targets and pathways that are not addressed by existing drugs and with potential to deliver deeper remission. Again, looking forward for partnership around novel targets and pathway with therapeutic potential that might address unmet needs in our core indications. And this is my last slide, and I think I almost over the time. And it's really the summary of what we, and very high level summary of what immunology emerging portfolio looks like now. We target key diverse pathway in innate or adaptive immunity, as well as in tissue mechanism with a strong link to our diseases, aiming at immune homeostasis. Our clinical asset represented here in pink. And I want to highlight that there is significant biology pathway overlap between respiratory and immunology, and we are maximizing with life cycle management in immunology the assets, which originally is respiratory assets. And in blue is our emerging preclinical portfolio. The portfolio is complementary to clinical assets, where we target new pathway and new modalities and aiming at multiple pathological triggers. With that, I would like to thank you for your attention and very happy to address questions.
Any questions for Tatiana?
Hi, Tatiana. Can you hear me?
Yeah, hi, Marina.
Hi. Tatiana, I wonder if you can tell us a bit more about your study or clinical trial with the anti-interferon receptor. And have you done any analysis? Because a third of the lupus patients don't have the interferon signature, which is clearly characteristic. So have you been able to dissect more about what happened in their patients, which clearly the clinical trial showed that they were not really respond as expected, that they were not responding?
All right. So, Marina, the situation is the following, right? TULIP-1 and TULIP-2 are the most recent study with anifrolumab. We do have samples, and right now we're processing the samples. So translational samples are not yet ready to be presented. We don't have a result yet. We do have results from the MUSE study, which has been run some time ago, and this is a phase 2 anifrolumab study. And the data from this study is published. A lot of the patients from MUSE-2 study had interferon high signature. Again, the study the translational study was published from phase 2. And phase 3 study is coming, and we're very curious to look at how the signature works in this study, what we can learn from translational samples in terms of not only interferon pathway, but beyond interferon pathway. What other pathways are regulated in lupus and not necessarily addressed by anifrolumab treatment?
Thank you. So there's a question online from Matthew. So he asks, in almost all of these diseases, there's clinical and translational response heterogeneity. So has the data from anifrolumab helped to understand lupus endotypes?
We definitely hope that it will help. Personally, and as a kind of bioscience discovery lead, I'm very much looking forward to explore the samples. One can think, right, that we can learn a lot in terms of how to treat these patients better, specifically when we look at partial responder or non-responder to anifrolumab therapy. Again, this is definitely my hope. The phase 3, both phase 3 is right now the data from phase 3 translational data are being developed. And we are definitely committed to take a look at this and do deep dive analysis.
Thank you. Thanks very much. So our last speaker before lunch is Charles Bangham from the Institute of Infection at Imperial.
May have to plug this in somehow. Try that one. Sunrise over Mount Fuji as I was flying in just before the pandemic. Thank you very much for the invitation to speak today. I'm going to be brief because it's just coming up to lunchtime. I'm going to introduce the Imperial College Institute of Infection, which we are just in the process of setting up. I often hear the remark from colleagues outside Imperial, for example, here in the Crick, that one of the real strengths of Imperial College is the fact that we have clinical medicine next door to excellence in engineering and physics and chemistry and maths. And we have the excellent potential to combine them in, for example, research on infection. And whenever I hear that remark, I think, do we really make the most of it? And in fact, we heard a moment ago from Ed Tate, outlaying his fantastic range of techniques and what they've done with their chemistry expertise. And you heard him say, we are looking for opportunities all the time to collaborate with biologists and medics. So this is what we aim to do. We do have, of course, a large collection of people who do research on different bugs in Imperial College. And so the aim of the Institute, which was just set up about a month or two ago, is to bring together anyone who's interested in any aspect of research on infection, who wishes to collaborate with people in different disciplines. So it's an interdisciplinary. And the aim is to identify the areas of overlap between these different disciplines and catalyze their interaction. Of course, this already goes on in Imperial, and a fantastic example is the Digital Diagnostics for Africa Network, which is between the Department of, among others, Department of Life Sciences, Department of Medicine, and the Department of Electrical and Electronic Engineering. So I think it's a truth universally acknowledged that we need more collaboration between the disciplines in order to accelerate this research. And so, as is summarized here, our mission is to catalyze this by bringing together the workers from different disciplines and to focus on specific aspects of infection. We have recruited a number of, as well as a steering committee, we've also have a series of champions which represent the different departments and faculties in Imperial College. Some of these you will recognize from having spoken today. Some have become media stars and continue to be, and some are both. We had our online launch just a month ago. We were very pleased with the attendance. We had more than 600 people registering for the meeting, and over the 2 days, over 450 attended at any one time. The opening talk, we were delighted to say, was given by Julia Gillard, here at the bottom left, former Prime Minister of Australia, and as you will know, now Chair of the Governors at the Wellcome Trust. And she gave an impassioned and really very effective short speech, indicating not only the need for the Wellcome Trust to align its strategy with infection. As you know, it's one of the three main pillars of their new strategy, but also the importance of the interdisciplinary work, which is championed by the Institute. And as you can see, we had good representation from the different faculties in Imperial at this launch. So just looking forward, what do we aim to do? We aim to build the community, to bring together the people by a series of not only seminars, but interdisciplinary workshops. For example, with the Grantham Institute of Climate Change in Imperial, which is very well established, been well there for 10 years. There's a lot of interest at the moment in the interaction between climate change and changes in susceptibility to an exposure to infectious disease, for example, the spread of arthropod-borne viruses. And so we're organizing a series of interdisciplinary workshops, bringing together the Institute of Infection with each workshop will have one other major discipline, for example, climate change. One of the major aims is, of course, postgraduate training, and we are aiming to launch an interdisciplinary master's in infection science. The point of this is to be almost like a MOOC, a massive online training program. That is to be interdisciplinary again, to be modular, to be mainly online, to have the ability to do it only online, and with a specific focus on retraining people who come from different fields. Some time ago, the Wellcome Trust had an excellent scheme for training mathematicians in biology. It's a mathematical biology fellowship, which unfortunately is discontinued, but it was extremely effective. And we have some people in the audience who strongly benefited from this. We aim to have an analogous program where we can train engineers or lawyers or people with any background who want or realize they need to acquire an expertise in infection to join our MSc program. And of course, we are aiming to launch a PhD program as well. We have already been approached by a large number of external partners in not only this country, but all over the world, from Australia, Sweden, Korea, South Africa, among others. And we aim to, again, catalyze the collaboration between Imperial College Infection Research and these other centers. For example, we have in a couple of weeks' time another workshop with our colleagues in Singapore in the National Center for Infectious Diseases. And we are going to be holding further workshops next year with, as I say, among others, the Grantham Institute for Climate Change. It also provides an excellent opportunity to strengthen what we all want to do, which is particularly to encourage early career researchers to stay in research and to advance their career and support them, and even as they do, as well as more public engagement and science communication. So as I mentioned, we've got the seminar series already launched, the workshops, and on the matter of science communication, we have an excellent collaboration that's just started with the program on the MSc in Science Communications in Imperial College, where the 7 students on this year's course have got a joint project on the history of infection research, not only in Imperial, but in the country. And next term, each one will be paired off with one PI in a different aspect of infection. So it's going to be really exciting to see what comes of this. You might think that this was a reaction to the pandemic, but in fact, we've been planning this for a very long time. In fact, I first suggested the idea some 14 years ago. It takes a while to get this accepted. But this is the future program. I won't go through all of the titles, just to say that in 2 weeks' time, we have our first meeting with potential philanthropic donors, together with the president of Imperial, Alice Gast, and about 60 or 70 people, some in person, some online, who are considering supporting the Institute. The aim is to raise between 50 and 100 million, and ultimately, we aim to have dedicated space for the interdisciplinary research. I should point out that it's dedicated space. We're not aiming to kidnap people from their existing department or faculty and bring them over into an institute and they leave where they were. They will retain their position. Well, what they do is collaborate with people in other disciplines, and that interdisciplinary project is focused on that in that space. So it's like a research hotel is what it's sometimes called. They retain their existing lab space and group, of course. So thank you very much for your attention. I think we are moving to lunch, and I'm happy to take any questions.
So we might start with this session. First, here with us is Professor James Moore from the Department of Bioengineering at ICL, and he's going to tell us about his approaches to understanding and modulating adaptive immunity.
Good afternoon, everyone. It's a pleasure to be involved in this wonderful symposium today. I represent the Department of Bioengineering at Imperial, where, as you can see in the box here, we have fairly large research operations spread across both of the main campuses, and a very large educational mission as well. We also, I should mention, have 4 entrepreneurs in residence who are very translational department. We are the first department at Imperial to even have an entrepreneur in residence, and now we have 4 of them. So I'd like to highlight first some of the fine research done by my colleagues. First of all, Reiko Tanaka, who's here in the audience, does some wonderful research on eczema, trying to understand not only the subcellular molecular mechanisms underlying the disease, but applying that in the true engineering sense to right on to diagnosis and treatment. We also have, I'd like to feature Dr. Sam Al, one of our younger members of staff, who applies microfluidics for various cancer applications. And all of this stuff on the this part of the slide describes a microfluidic device he designed to separate T cells and optimize them for immunotherapy. And that's now been licensed to a company in the Netherlands. So I think showing these 2 slides gives you some idea of both the computational, mathematics-based computational expertise that we have, as well as the experimental expertise. That's something you'll see as a recurring theme, not only in my work today, but throughout my department, and something that we're very proud of. So I'd like to tell you today about the work I've been doing in the lymphatic system in the direction that in so much as it applies to adaptive immunity. And I'd like to describe that in the framework of giving someone a vaccine. So when we give someone a vaccine, it has the antigen information in it. It may also have an adjuvant that's injected typically intramuscularly, although there are more initial lymphatics in the intradermal space, subcutaneous as well. So that information needs to be taken up by the initial lymphatics, transported via these the pumping action of these larger collecting lymphatic vessels here into a lymph node, which might be several centimeters away. And then once that information has come into the lymph node, either as captured by an antigen presenting cell or in solution, then that information needs to be delivered appropriately to the correct cells within the lymph node. And as you're aware, the lymph node is a highly compartmentalized structure. It's not just an amorphous bag of cells. It has specific B cell zones and T cell zones. So that's step 1, is to take up the information into the initial lymphatics. Step 2 is to get it pumped to the node. And then step 3 is to transport it within the node. And I think the right way to look at this is from the node's point of view. What temporal profile of information delivery is required to optimize adaptive immunity? And that's sort of the central question of what I'm going to be presenting today. So let's start with the transport through prenodal lymphatics. This is what the pumping action looks like. This is a view of a lymphatic vessel doing one of its most important jobs, and that is returning fluid from the periphery centrally to the blood system as a matter of fluid balance. So you can see several cells suspended inside this lymphatic vessel. Those are lymphocytes or dendritic cells. And so we can track the velocities of those cells flowing through the lymphatic vessel and also track the diameter of the vessel. And what we find out is that those cells are traveling at a velocity on the order of about 1 centimeter per second, and that the flow rate is about 200 microliters per hour. And yes, I'm proud that we can measure flow in microliters per hour. But I'd like to come back to this velocity, 1 centimeter per second. Now, if you need to deliver antigen information from the periphery into a lymph node, I can tell you this is the fastest way for it to happen. Even if an antigen presenting cell is crawling, which it does, crawls towards the initial lymphatics once it captures information, to get it to the lymph node, you need it to ride along in this flow stream here. It can't crawl all the way to the lymph node. The velocity of a crawling cell is 8 orders of magnitude slower than the flow of this fluid here. So it's something like comparing the speed of a walking human to the speed of light. Now, I should also point out this spinning image on the right here is a confocal stack of a valve. So there are valves just upstream and downstream here. So lymphatics do their job of pumping with a combination of squeezing and then these check valves, which are quite closely spaced. So as we become more interested in the effects of different substances on lymphatic pumping, we've actually been working on both computational modeling and experimental work on lymphatic pumping for a couple of decades now. And we started to look at the effects of vaccine adjuvants on lymphatic pumping. And so we know that adjuvants help. So we with adaptive immunity, actually, they slow down lymphatic pumping. It's not what we expected to see. It actually works something like nitric oxide on lymphatic vessels. So it relaxes the vessel in its diastolic state, and it slows down the frequency of contraction. So it actually is kind of shutting down lymphatic pumping. And I think the natural question is, well, why? And why do vaccine adjuvants help if they slow down lymphatic pumping? And the truth is, we don't know yet. What I'm showing you is unpublished data. We're still in the very early stages of trying to figure out what vaccine adjuvants actually do. So I'd like to move now to talking about transport within the lymph node once the information comes in, and either again in the form of captured by an antigen presenting cell or in solution. So there are a few different chemokines that are important for cell migration in lymph nodes. We have been focusing on CCL19 and CCL21, and they set up concentration gradients inside lymph nodes, and cells migrate in the direction of increasing concentration. And the actions for setting up those chemokine concentration gradients span both the biological and the physical. So CCL19 can bind to CCR7 or ACKR4, so can CCL21, but CCL21 can also bind to extracellular matrix. So you got a few different biological interactions going on there. There's also diffusion. So these are fairly small molecules that can diffuse through the interstitial space. They can also be transported by lymph flow. So I'll show you what lymph flow looks like inside a lymph node. Now, this is not something you can measure. We did our best experimental attempts to quantify the things necessary to construct a computational model. So this is the results of that computational model, showing you the lymph flow velocities, mainly coming around the edge here, but some of the lymph does get transported into the middle of the lymph node where it goes out through the walls of the blood vessels. So those flow patterns are important for setting up those concentration gradients of these chemokines. And I should also point out that migrating cells can also affect the local chemokine concentration field as they migrate through the interstitium. So we set up a computational model then based on that fluid flow model of the chemokine transport inside the lymph node. And again, one of the things that we can do with computational models is show you things that can't be measured experimentally. So these are concentrations of unbound CCL19 and CCL21. And I'll just note that these are singular digit nanomolar concentrations. Now, the bound CCL21 concentration is much higher. It actually can be measured experimentally. It's a couple of hundred nanomolar. And there was an experiment published a few years ago which showed that antigen-presenting cells will migrate up these concentration gradients in this interfollicular zone between the B cell follicles right here. And you can see that changing color indicates that concentration gradient. So what these authors did was they showed that knocking out the atypical chemokine receptor for these chemokines disrupts the migration of those cells. Now, atypical chemokine receptors are not associated with migration anyway, so this was a bit puzzling. And so what they, I'll just go back here, what they decided the effect was that in the lymph node capsule on the ceiling, you have these atypical chemokine receptors, and they're sucking away the chemokine from the parenchyma of the lymph node and setting up that concentration gradient. So we got suspicious of that finding and started modeling it. And actually what we found was they did another knockout that they were not aware of. They knocked out the lymph flow in this thing. So as you may be aware, when you do immunohistochemistry on a lymph node like this to look at chemokines, you have to cut it out, process it for a while. And so when you cut it out, you've actually shut off the flow. And then we can model this computationally. And what we showed was that you need to shut off the flow and the ACKR4 to reproduce the experimental result of abolishing the chemokine gradient and interrupting the migration of those cells. So I think this is a nice demonstration of how our approach of combining both experiments and computations can really bring insight that you can't get from one or the other. So let's come back to this picture here. So again, step one is to get the information in. Step two is to transport it with the pumping. Step three is to get it to the right place in the lymph node. And then there's this question, what's the temporal profile required to optimize adaptive immunity? And I think that's where this process ought to start. We ought to start with the lymph node itself. Ask the lymph node what it actually needs in terms of an information delivery profile, and then come up with vaccine adjuvants that produce that by their modulation of the pumping lymphatics and also the way they're taken up by the initial lymphatics. So let's focus then on the what the lymph node needs, and then use that as a rational quantitative basis in the engineering tradition as a basis for adjuvant design and other aspects of modulating adaptive immunity. So that's kind of where we're headed with this thing. I wanted to give you not only a flavor of what we've done before, but what we plan to do in the future. And here's is my summary of what I've told you today. And again, what we would like to establish is what does the lymph node need and how do we deliver it? So with that, I'll close with acknowledgements of the people that helped out with this research, both collaborators and colleagues at Imperial, as well as funding sources. And I'm happy to answer questions.
Can I just ask you, so would nitric oxide itself then be a great adjuvant?
I'm not sure it's ever been tried. It certainly does instruct, like blood vessels, it instructs lymphatic vessels to relax. Actually, lymphatic endothelial cells produce copious amounts of nitric oxide. It's transported within the lymphatic vessels, and we've shown that it gets kind of stuck behind the valve leaflets, which we think is important for cellular signaling as well.
And if I may, a second quick one. So one of the adjuvants you showed is an emulsion. So can you play with emulsions to change these characteristics? Because you can't play much with alum or CPG as such.
I'm sure you can. Perhaps our colleague from GSK can tell us how much they've been playing around with it. But I think previously it was a big black box between coming up with an adjuvant and seeing what effect it had on antibody counts, for example. And I think we need to figure out what's in the black box, and that's really our goal. Thank you for the question.
So that was very interesting. So I was wondering to what extent you are also modeling the fact that both the afferent lymphatics and the lymph node are changing during that spatiotemporal course of transmitting the information to the adaptive immune system. So we know there's relaxation of the follicular reticular, of fibroblastic reticular cells that can expand the node, which presumably alters fluid flow. And there's also the whole conduit system that can provide a sort of shortcut for chemokines to get quicker down into the node. So I'm sure you're thinking about all these things. I'm just wondering how you're trying to model those in.
Yeah, these are topics close to my heart for sure. So one thing that we've done, which I didn't have time to show today, is we looked at the effects of a couple of adjuvants, the toll-like TLR activators, on lymph node flow patterns. And so what we find is that, you know, within 24 hours, the node has started to swell, as you allude to, and it's still swelling after 72 hours. And what we've done is we've measured the flow resistance across a lymph node. So, I mean, these things are mouse lymph nodes, are very tiny, so it's an exquisite experiment. But what we can do is we've shown that the flow resistance of the lymph node goes up by an order of magnitude within 24 hours, but within 72 hours after the vaccination, it's gone back down to its baseline flow resistance while the node continues to swell. I think there are some incredibly interesting things going on there. We don't know, we haven't solved all of the mysteries of what's going on there. I think the other interesting thing, which we didn't have time to talk to, but we've done a lot of work on, is the lymph node blood vessels. Fascinating structures. You don't see them in other parts of the body, the high endothelial venules, and there's a lot of interesting exchange of not only fluid, but cells across those structures.
Okay, so we are going to, thank you very much.
Thank you.
James. We are going to move to the next speaker. And this is Professor Claire Lloyd from the National Heart and Lung Institute at ICL. She's going to talk about immune regulation in the airways.
Thank you. So I'm going to talk about those of us at report on the work that those of us in the National Heart and Lung Institute are doing, looking at immunology of the lung. So if one thinks about the lung, it's kind of complex place for the immune system to cope with, in that every time we breathe in, then we're breathing in a whole range of potentially antigenic material. And yet our immune system has to distinguish between those that are potentially harmful, so pathogens such as viruses, fungus, and bacteria, and then discriminate those from innocuous particles. And for the most part, most of us are tolerant to these, to things like animal danders and pollutants. But when tolerance is lost, that can contribute to quite devastating lung pathology, which compromises lung function. And so immunity in the lung is organized in between both resident immune cells, like this airway macrophage, which is sitting in an alveolus, as well as recruited lymphoid cells, which are recruited to the lung after signals given by resident cells, but also those cells which traffic through performing immunosurveillance. And they work in concert with a range of different stromal cells which are resident in the lung, and we're particularly interested in these epithelial cells which line the respiratory tract and actually are intimately involved in the direction of immune responses. And so these interactions between the sort of lung stromal cells and immune cells are influenced by a wide range of both intrinsic but also extrinsic factors. So we know that our infection history has an impact on our ability to respond to another infection. Obviously, allergen exposure into those people who are atopic is really important in directing severity of asthma reactions. We know that genetics and drug use, so not only inhaled corticosteroids for the treatment of lung diseases, but also things like sequential antibiotic use. The environment that one's reared in, so whether in an urban or rural environment, is important. And we know that smoking is a risk factor for multiple lung diseases, as is our level of nutrition. So obesity and poor nutrition contribute to severity of different diseases. We know that the microbiome informs these immune responses, and not only the gut microbiome, but the local lung microbiome also has a really important role to play. And one thing that we're very interested in as a group is the immunity across the life course. We know that the immune responses within the airways are actually very different in the very young and the very old, particularly when I'm talking about respiratory infections. But we know that there are multiple lung diseases which are actually age as a risk factor. So things like pulmonary fibrosis and COPD. So we have a range of animal models of these lung diseases. So we have models across the life course, so neonatal, but also age models. The lungs are pretty accessible, so we can deliver agents directly to the lungs, so we can cause allergic immune responses or infections with viruses, fungal particles or bacteria, but also pollutants such as smoke or toxic agents such as bleomycin to promote pulmonary fibrosis. And in order to manipulate pathways, we use either reporter mice or specific cell-specific deletion, either in immune cells, but also there are a range of different Cree drivers which allows us to control gene expression in resident lung cells. And we've also taken advantage of different viral vectors which allow us to overexpress particular genes within areas of the lung, here shown expression in the bronchial epithelium. And the analysis of these models is general lung physiology. So we perform multiple different parameters of changes in lung function, pathology and imaging, high parameter flow using Aurora and also Vitessa, transcriptomics, metabolomics and metabolism, and also analysis of microbiome. But all these animal models are, as Marina mentioned earlier, they're great to be able to manipulate different pathways, but we do all this in the context of a translational research program. We're a mixture of basic scientists and clinical academics, both from the Brompton Hospital, but also from Imperial hospitals, where we're lucky enough to be able to get samples from well-characterized cohorts of patients. So as I said, the lung is fairly accessible, and particularly at the Brompton, they have a very well-organized and active bronchoscopy program, which allows us to collect cells from the airway lumen. So those cells which are resident within the external, so they're in contact with the external environment. And so we're using a washout, we can collect cells, count them and phenotype them, both in terms of flow, but also transcriptomically. And then we can collect samples from this airway wall by brushing, and then culture those cells here, just showing how culturing those cells ex vivo at air-liquid interface, but also via endobronchial biopsies, which allows us to do immunohistochemistry and more complex analyses like Hyperion. And more recently, in order to try and look at multicellular interactions, we've used human lung resections to either isolate different lung cells and grow in vitro, but also to image cellular interactions using precision cut lung slices. So on the and I think an example of this, one of these studies is a study that was done collaboratively to James Harker, me, Pallav Shah, who's a clinician at the Brompton and the Chelsea and Westminster Hospital, and Jimmy Peters, where we looked at a cohort of patients who had recovered from COVID, who were hospitalized but then recovered but still were symptomatic. And Pallav had a bronchoscopy program to isolate cells from those patients. And so we're able to get blood and bowel and do high parameter flow on them, as well as proteomics, and then correlate those immune features and proteomic features with markers of lung function. So I just sort of highlight a couple of the specific areas. I think one of the areas I want to highlight is Professor Sejal Saglani, who's an academic pediatrician. And she has a cohort of patients at the Brompton where she's looking at progression from preschool wheeze. So these are children who are wheezing with viruses or other or non-specific triggers. And we're trying to understand why and how they progress to asthma. And this is a bronchoscopic program as part of their clinical workup. They're really exquisitely phenotyped, so we know that they're taking their treatment, but they're still symptomatic. And by collecting a whole myriad of different clinical and immune markers, she's been able to in collaboration with Reiko, to do cluster analysis and identify different phenotypic clusters, which we can then model in our neonatal mouse model. We're very lucky in that part of this clinical workup is the sampling from both the airway lumen and also the airway wall via endobronchial biopsy. And we're at the moment trying to analyze how the airway wall differs in a preschooler with wheeze, a severe therapy-resistant asthmatic, and then a normal child. Adam Byrne is a senior lecturer in the department, and he's really interested in where macrophages come from and how immunometabolism dictates how the lung is able to repair itself. And so he's he had a collaborative program where we looked at how macrophage ontology in humans detects function, and that was done across the life course. He's has a number of different studies looking at how macrophage metabolic products influence lung environment, and showed that a metabolic product actually really involved in the recovery or development of idiopathic pulmonary fibrosis, and looked at how different metabolic phenotypes affect lung inflammation, showing a role for iron and also the DNA methylation pathways in patients with chronic pulmonary fibrosis. And then looking at transcriptional regulators such as IRF5 in both IPF and also asthma. James Harker is also a senior lecturer in the department, is really interested in understanding regulation of antibody responses in the respiratory tract, again across the life course. So he's using both pediatric and adult patients, as well as mouse models of different ages, to look at how wheezing, allergen sensitization and respiratory infections and how the protective immunological memory is developed in those children and mice, looking at the role of both B cells and resident memory T cells. Rob Snellgrove is a professor who's interested in how the lungs repair themselves. So I think he and I are both involved in how chronic inflammation contributes to lung repair, and how one can look at the normal response to either infection or lung injury is a program of repair and then resolution of injury and a restoration of homeostasis. And that's very important in the respiratory tract, because any constant remodeling will affect the ability of the lungs to function. And for the most part, this is very successful, and there's restoration of homeostasis. But in a proportion of patients with different signals, so that signal can be inflammation or it can be toxins, there's persistent inflammatory responses, and then tissue remodeling, and there's an abnormal deposition of matrix. And Rob has been looking at how matrix components differ between different diseases. And he's outlined a pathway mediated by leukotriene A4 hydrolase in directing different lung diseases. This is a really interesting pathway that has both pro-inflammatory and anti-inflammatory responses. And he's been able to generate, to divorce the different functions of this one molecule, identifying a matrix fragment, PGP, which has effects on neutrophil recruitment, but also quite differing responses in terms of epithelial proliferation and repair versus fibroblast proliferation, and outlined that with this one matrix fragment, if you've got too much PGP, then that's associated with COPD and asthma, but too little PGP is associated with chronic repair problems and remodeling, such as IPF. And he's got a drug development program actually looking to develop small molecule inhibitors, which target just selected actions of LTA4 hydrolase. So my own program is also interested in looking at repair and looking at how the lungs sense the external environment. So I said that we're breathing in pathogens and pollutants and allergens, and I'm really interested in what cells recognize these signals as either dangerous or innocuous and should be ignored. And we've previously done a lot of work on the epithelial cells, and now we're looking to see how these epithelial cells, in concert with the neuronal pathways, signal to the cells in the underlying mesenchyme. So those can be fibroblasts, airway smooth muscle cells, but also cells of the immune system. I've got a satellite lab here at the Crick with Anne O'Gara, and we're doing some transcriptomic analysis of these epithelial cells. And through that, it's led to a collaboration with Vasilis Papnis, who we're working with to look at lung neuronal pathways using some of his reporter mice. So from previously doing transcriptomic analysis are really interested in actually what the cells look like in situ, and so have developed different methods of imaging those lungs. The slides will move forward. This has been this is a precision cut lung slice where we're looking at ILCs in green and T cells in blue, and just looked at the comparative movement patterns of different cells, and found that actually movement of cells is really affected by the local tissue matrix, and affects their ability to then contribute to inflammatory reactions. So from those mouse studies, we've realized that it's actually really important to understand which cells are within the airway wall and who they're talking to. So we're working with a Hyperion machine, which is in the Department of Brain Sciences, to actually look at different immune cells which are in the airway. So this is a patient where we've got tissue from interstitial lung disease, and this is a pediatric asthma tissue where we're just working out different ways of looking at both stromal cells, matrix, and different immune cells in samples from patients. And the goal of the program is to integrate this imaging mass cytometry with spatial transcriptomics, and then looking at matrix geometry by second harmonics. I'm going to leave it there. Just finished. Thank you.
We made you rush. Any questions for Claire? How good are these animal models when you were talking about the asthma, the neonatal asthma? How
the neonatal asthma models actually reflects certain aspects of the pediatric disease. Obviously, we're trying to measure model particular aspects of disease. So, but I'd say my clinical colleagues are quite robust in their dissection of our models, and so we make sure that they look or they reflect particularly the pathological and physiological aspects of the disease. So particularly the neonatal model, we've been able to map different inflammation and repair pathways, and the ones in the mouse look very similar to the ones in the children.
Great. Marina?
Claire, nice. I just want to ask you, do you think there is a kind of way in which the signal from the lung is then transferred to systemically in a way and make the person then to maybe overcome their asthma, which is quite typical in children to overgrow the issue of the problem, and then developing maybe some other genetic predisposition to other diseases in a different organ? How do you think of the environment of the lung then determine maybe something else that happen when you are old?
Well, certainly, I think Rob's got some really interesting data showing that when we can look at inhalation of allergen as affecting, hematopoiesis very specifically, depending on the antigen. So you can see, we knew that that's what happened to eosinophils, but actually there's a real effect on neutrophil-apoiesis, and then also recruitment of innate lymphoid progenitors. So I know that there is a signal that comes from the lung going systemically. A lot of asthma is genetic, but it's also gene by environment. So I think that we think that there is a kind of perfect storm where you've got the right genetics, the right environment, maybe multiple viral infections, and then that drives pathology, which then culminates in asthma. And I think the asthmatics that we're talking about, the ones that we actually bronchoscope, they're really severe therapy resistant, and they unfortunately they don't actually grow out of their asthma. But when we're looking at the preschool wheezers, then most of those do grow out, but we just want to know what the difference is between someone who is going to go on versus someone who will recover.
Thank you very much. Thank you, Claire. So we're going to go on to our next speaker, who is Dr. Christoph Burkhardt from Novartis, and he's going to talk about discovering medicines to improve lives of patients with immunologic diseases.
And it should come up in a sec. First, let me thank the organizers for choosing and offering the opportunity to Novartis to present our work.
love to see that you see more than just the coast of Northumberland. Because I see it on my screen. How comes?
We have some IT support coming. Thank you.
So, if we do that, fire it up again.
Here we go. Excellent. Thank you very much. Thank you much. So again, let me thank the organizers for being allowed and having the chance here to present some of Novartis in a nutshell. Novartis is a large organization with about 100,000 employees. So you can imagine that this is a huge task. Also, what you see here, I'm working since 20 years at Novartis as a project team head and lab head. 3 years of it in Vienna, 17 years in Basel. I'm not the director of, but a director at Novartis. And that is the Novartis Institute for Biomedical Research, NIBR. That's our research organization. I will then give you some more information about it later on. And there it's a subdivision, a so-called disease area, ATI. ATI stands for autoimmunity, transplantation, and inflammation. It's essentially our immune research part with about 200 employees. And so just also a little bit as disclaimer, we are working also like the colleagues in academia that you just heard, and also like our industrial colleagues with the patient in mind. So it's essentially all the work what we do basically from starting off with conceptualizing a drug, identifying a target, developing a drug, finding it, and then bringing it to the clinic and eventually bringing it to the patient. We do that with the patient in mind, trying to understand the way of the patient, the journey of the patient in his or her disease, and trying to alleviate any issues that the patient will have. And this we do in quite a lot of countries. You see that Novartis has about selling its product in about 155 countries. We have been reaching in 2020 almost 770 million patients. The target in the near future is actually to reach up to 1 billion patients worldwide in a year. And we also have dedicated programs to give better access to healthcare and also to medicines for people in need. That's especially our malaria program, for example, where we reach about 66, or where we have reached in 2020 about 66 million patients. Now, at the center of the organization, headed by Jörg Reinhardt and the CEO Vas Narasimhan, everybody calls him Vas, is our research and development organization, which is essentially two parts. One is what I mentioned already, NIBR, Novartis Institutes for Biomedical Research. They're doing the early part of the research up to the first proof of concept studies. And then we have the GDD organization, the Global Drug Development Organization, which does the bulk then of the clinical studies up to the registration. You see the numbers, net sales and net income, but that I think we have some more numbers here, and more numbers is that our R&D, that is NIBR plus GDD, we invest about 18.5% of our annual revenues. That's $9 billion US. And we have about 160 pipeline projects here from phase 1 to phase 3. And 90% of those we aim to be first in class or first in indication. And we had, again, in 2020, we had about 26 major regulatory approvals in different countries, different regions of the world. Now, NIBR is an organization with about 5,600, varies slightly, employees.
scientists, physicians, and business professionals, headed by Jay Bradner. You may have seen in an earlier talk, you may have seen one of his compounds, a degrader compound, because that was in his academic life, was where he was gaining a lot of reputation and experience. Now, since a couple of years, he is the president of Novartis. Now, we are a global company where the research is split over several continents, with a core in the US, in Cambridge, Massachusetts, a big center in Basel, and where the headquarters of Novartis are, as well as a center in Shanghai. We have roughly 300 early development programs, and I mentioned ATI as one of our disease areas. We have 9 disease areas, and these disease areas you can see here, and on the right you see how the projects are more or less distributed across these disease areas. The ATI is here in light blue, the autoimmune transplantation information, essentially focusing on diseases, immune-related diseases. Transplantation was at the core of it, but it has slowly phased out, or is in the progress of phasing out. We still have transplantation programs with an anti-CD40 antibody ongoing, but essentially now the core is rheumatologic diseases, is dermatologic diseases, autoimmune indications, and inflammatory indications. And then you see a whole range of other disease areas. You see the immuno-oncology here, that might be of relevance to some, and then also the oncology, neuroscience. And we had dedicated institutes also. We had an institute for tropical diseases, once in Shanghai. It has been relocated to Cambridge, and you see here the neglected and tropical diseases that is run by that. We used to have an institute, a respiratory institute in Horsham, close to Gatwick Airport. That has been integrated also into this disease area in Cambridge. So that's now the respiratory disease. You see here, DAX, more or less. So I'm in ATI, and so what I just mentioned before, so what NIBR is doing is from conceptualizing a disease progress, a pathway, trying to find a target, and I think this is all things that you're very much familiar with, a target that has any relationship to the disease progress. We need to validate the target, we want to validate the target, and then comes the long way of finding modalities, therapies. These could be biologics, these could be classically low molecular weight molecules, to inhibit that target or to stabilize it, even to increase that target depending on what the pathway is telling us. And then finishing, and this has been a concept and Novartis is running about since 15 years, then in a small dedicated patient population, do a proof of concept. What we need to understand is in that patient, what the target is actually doing in that patient population. And the better we understand, of course, the disease and the role of the target in disease, the easier it is to get this positive proof of concept. And then from then on, basically have to go for a further larger phase 2 trials or then phase 3 trials. Now, the whole thing is a marathon. It takes about 10 to 15 years to come from up to 10,000 compounds. I worked on the project for 15 years, and in my lab, we basically have the first compound. Now we have just concluded the phase 3 study. 15 years is quite a normal time. We wish and we have efforts to make it faster, but then in our case, we started off with about 15,000 compounds and ended up with one single clinical candidate. So it is quite an endeavor. And it also, it can be for a scientist quite a frustrating thing, because most of the 200 or so associates and scientists in ATI, they don't see the light of their project. About 5 of them will see that their project will continue to be tested in man, and maybe 1 out of the 200 will see the project and the compound eventually registered. Many of them retire before they see that. So it's something that where you need to have a certain frustration tolerance, and failure is the norm and success is the absolutely exception. Now, when we When we look at the earlier stages and basically the steps we go through from the target validation, I mean, basically demonstrating that a particular molecule, a particular structure is relevant in the disease, and modulating it, changing it would alleviate and change the disease. Then from assay development, where we can build systems, usually in vitro systems, then also in vivo systems, where we can demonstrate that manipulating that will have a benefit on disease-like parameters, usually in a mechanistic setting. And then there's a long path of the medicinal chemistry, an iterating process where we always go from a poor compound to a better compound, that one that fails maybe due to then the next step, preclinical safety due to tox, and go back to square one and modify again and modify again. Until out of these many molecules, we come to one that actually we think it's good enough and safe enough to be tested in man. And then we go to the proof of concept trials in those patient populations. Over the past 15 years, these are a number of compounds that have actually made it. They have made it to the market. They have made it to the patient. The ones that are encirculated in blue are ones that originated in ATI. And some of you may be familiar with you. So we have this group Mayzent and Gilenya, these S1P receptor agonists, and we have them in multiple sclerosis and different forms of multiple sclerosis. We have Ilaris, which is an IL-1B inhibitor or antibody in the CASP indications, but also then used in many other different indications. And then finally, we have Cosentyx, the anti-IL-17A antibody, very successful treatment for psoriasis and a number of diseases such as psoriatic arthritis, ankylosing spondylitis, etc. So but then you have many, many more. But these are the ones where ATI was significantly involved. So what we want to have new science for new therapies, and we want to go to the area where things are not druggable or not have been considered not druggable before. And we do that through, for example, modalities and platforms that cell therapy, biogene therapy, radioligand therapy, biotherapies, quite a lot of those. We employ more digital sciences and digital technologies and AI also for shortcutting and making the clinical trials more informative. And I think what is then important is the collaboration. Like when England beat South Africa on Saturday, it's a team sport. So we need players. We need a good team. Here you have a lot of those, about 300 academic and about 100 industrial collaborators that Novartis has. And also it has been rated in the Nature Index as one of the main contributors in the world across. And I think what is particularly important for this is that we have this NIBR Global Scholars Program. I've even heard in the conversation that some researchers here have actually applied. This is like a grant. It's a competitive grant that started in 2020. About 700 proposals were received. And it's meant to fund for 3 years for up to $1 million research that is otherwise not funded. And in last year, 6 investigators in the projects got this type of funding. We had another round, and we just had the final round of applications and selections this year in 2021, and we will have, I'm sure, another one in 2022. And I think with this, we also have a number of postdoc positions and internship programs. So there's a lot of things also for young scientists where the industry, academic collaboration and partnership can continue. And with this, I'd like to thank you for your attention.
Thank you very much. Do we have any questions?
I have a quick one. You can hear me.
Yes.
You do it from the microphone.
The 6.
The 6 investigators that you mentioned were founded in the last round of the NIBR. Which countries and which topic did you found in that occasion?
Actually, I don't even have that information. But as we had a lot of US applicants and we had a lot of European ones, also Asians, hardly anything from Africa. So my guess, it would be a bit like the scientific community is distributed. We would have probably 3 US and 2 European and 1 in Asia. But I have not. I can find that information for you, but I don't have it. Yeah. I know of UK applicants, but I don't know whether any of the UK applicant really got it.
Marina.
Hi, very nice. I'm just going back to your frustration of the 15 years of work to get one compound. So the question is, how can we reduce that frustration? Because it is clearly a shared frustration, and the coronavirus has shown us clearly that when we need it, we can cut and get to the same point in a much faster way. So I know it's it's not just directed to you, it's directed to everyone, but how we can make that frustration less frustrating.
I mean, that question of how can we reduce the time in the early as well as in the late phase of drug discovery and development, this is an old one. It's basically as old as that. And for example, as when then large library screens, automated screens, or when the genomic information became available years ago, one thought that this will really gives us shortcuts. Each animal model was meant to give us a shortcut of informing us earlier whether something works or not works, or whether something is toxic or is well tolerated. Unfortunately, this hope has not really helped. And I think COVID and the mRNA vaccines in particular are a fantastic example of possibilities. There we have to compare, of course, the traditional way of doing vaccines. And many things were just basically the stars were lined up, and there was a lot of additional money, additional synergy, additional collaboration, and will to do that. Now, to learn from that and translate that into normal drug discovery, I think that is what we would like to do, and it's a great challenge. But I think there must be possibilities to do that. And of course, doing cleverer clinical trials is already something. Having better patient selection is another one. And it depends very much on which disease indication you work in. Sometimes you just need hundreds of patients, and you need a long time until you see a difference between a treated and non-treated group. Sometimes you can see it in a few weeks, and that will also help. So going usually there where you have the quick effect before you go into big studies with long-term, long studies, that's also one way of making it shorter.
Okay, thank you very much. We will now go to our next speaker, who is our local speaker, Gita Stockinger, and she's going to describe immunology here at the Crick.
Right, you may have noticed that I'm the odd one out, the only Crick speaker, with the heavy responsibility of representing immunology at the Crick. And I will give you a blitz tour through immunology, which is not totally comprehensive, but there are many of my colleagues in the audience, so capture them if you want more detail later on. I hope I'm coming on. I was told not to press any buttons, it would be there.
We know they're looking into it. I saw some action.
Adam is looking into it.
Someone is coming to the rescue.
No. Mhm. Great, thank you. Well, I can see it. There it is. Okay, immunology at the Kirk. So I've looked through. I mean, immunology, as you know, is a bit like metastatic disease. It really invades all kinds of disciplines. So it's not that easy to say what actually is an immunology group and what isn't. So I made a rough calculation. We have around 11 hardcore immunology groups here. We have groups that study the immunology of infection. We have quite a few groups that are mainly studying infection, so they focus more on the pathogen than on the host response to these pathogens. I left them aside for the selection today. I just want to mention one group who really sits in the middle there. It's Jean Langhorn's group, who works on the immune response to malaria, and she has about as much focus on the peculiarities of the pathogen as she has on the immune response against malaria. And we have also several groups that focus on cancer immunology, as well as some more unusual outliers, and I mentioned at least one of those. So to start with, as I said, it's a blitz tour. Victor Dubulovich's lab at Crick. Victor has, throughout his career, mainly focused on signaling in B cells. And in the course of his studies, he's currently focusing on a particular kinase, the Wnt kinase, and its regulation of adhesion and migration for B cell responses. And he also has an additional program together with Lizzie Fisher at UCL, looking at the genetic and cellular pathologies that cause Down syndrome. So quite divergent programs. We have the group of Veni Papagiannopoulos, who is focusing on neutrophils, a group of immune cells that is now much more in the focus, but had been sort of sidelined for quite some years. And his specialties look at the tuned immune responses by neutrophils depending on the nature and the size of pathogens. There was one publication a couple of years ago where he showed that neutrophils can sense the size of a pathogen. Those that are phagocytosable, they will deal with in that way, whereas large pathogens that they cannot engulf and phagocytose, they deal with by so-called netosis, by spitting out their chromatin and killing pathogens basically extracellularly. And on the left, there's a whole list of potential triggers for neutrophils, which they handle in different ways. I just want to, I haven't got a pointer, I hope you can follow this. Crystal neutrophils have been shown to have an important role in atherosclerosis. They actually recognize cholesterol as a danger signal and commit netosis in response to that, subsequently licensing macrophages for the release of inflammatory cytokines like IL-1. Neutrophils also play an interesting role in sepsis, when he looks particularly at fungal sepsis and described certain abnormalities of neutrophils in this setting, which also became quite obvious in COVID, where he was working on. Andreas Wack group continues the theme that Claire alluded to. He's interested in lung epithelial differentiation and repair, initially all in the context of influenza infection, but he now also has COVID models. He has studied the role of type 1 interferons both in antiviral defense, but also in their role of pathology, which happens in imbalanced situations, and their impact on the repair process. And he's interested in long-term changes in lung macrophage function as one of the side effects and long-term effects of lung infection. And so it's epithelial differentiation and repair at the heart of his program. George Kasiotis is best described as a retroviral immunologist. So he is interested in immune responses to both exogenous and endogenous retroviruses. He was very much engaged in the setting up antibody tests for detection of coronavirus responses. So this initial flow cytometry-based test that had then been adopted by the pipeline had been set up in his lab. And George is also interested in exaptation of endogenous retrovirus. I have to look this up on Google, actually. Both in the context of oncology. So, for instance, there are pro and anti-tumorigenic effects. He has described endogenous retrovirus as generating neoantigens for recognition by the immune system in the fight against cancer. But he has also described a way of how endogenous retrovirus regulate the release of soluble PD-L1, which might be a counter argument to that. Just in general, there seem to be an incredible number of physiological functions that are facilitated by the action of endogenous retroviruses. And George has his eye on many of them. Adrian Hayday has a lot of programs, but the main program here at the Crick focuses on an unusual type of T cells, so-called gamma delta T cells, which are particularly important at barrier surfaces in the body, such as the skin and the gut. And he has been following them from their generation, development in response to particular gene families expressed by epithelial cells, such as the butyrophilin family, which are important both in shaping their development, but also maintaining these cell subsets. And he is very much interested in what these particular gamma delta cells recognize. They respond to stressed epithelial cells, which could be markers of malignant transformation, and most likely these particular gamma delta cells play important roles in the very early, more or less innate recognition of such epithelial alterations. He also has been part of a huge consortium, the 3I consortium, characterizing mutations for their impact on immunology and infection. And he has played a big role also in COVID times by, for instance, setting up a proteomic pipeline to streamline severity of COVID-infected patients and for characterizing immune cell responses in the course of this infection in patients. Caetano, who's sitting here and with trepidation, I must say, it's difficult to summarize his program because it's incredibly big. But the center emphasis in Caetano's program are dendritic cells, who themselves, of course, are incredibly versatile, play a role in the initiation of both innate and adaptive immunity. He has done seminal work on the ontogeny of dendritic cells, working on the role of dendritic cells in cancer and in their recognition of cytoskeletal dumps, which is very with actin in center stage here. And on this particular DC-expressed molecule DNGR1, which characterizes a subset of dendritic cells that are particularly important in cross-presentation and as such in recognition of tumor antigens. Not to forget the whole set of work on the recognition of viruses in this setting. Dinesh Kalado is a link between immunity and cancer. He's interested in B-cell development and it has focused a lot on the germinal center B-cell reaction, plasma cell and memory B-cell differentiation, and the relation to lymphoma and multiple myeloma in this setting. And then the slightly unusual exception I wanted to present here is Vasilis Paklis, who already appeared in Claire's talk. He's really a developmental neurobiologist, but he's very interested in the interaction of the enteric nervous system in the gut with the immune system. And in a recent paper, he described an important role for enteric clear cells in regulating re-establishment of homeostasis by responding to gamma interferon, recruiting CD8 T cells, and setting up the first stage of tissue repair in a type 1 immune response before the general type 2 immune response in this model. It was a Helminth infection that, of course, causes a lot of damage in the tissue. Before the type 2 response, there's this particular type 1 response which involves these clear cells. Anu Gara's group here is, of course, historically interested in the regulation of IL-10 production by different types of immune cells. That cytokine is made by many immune cells, but is regulated in different ways. And her other focus in the lab is understanding human TB, and in particular, latent disease and the factors that make it either maintain as latent disease or progress to active TB. She does this both on mouse models and in human disease, collaborating with hospitals in Leicester and using a lot of transcriptomic analysis from blood and lung samples to measure the progression of this disease. Then we have Patricia Barral, who is a secondment from King's College, whose focus is NKT cells, a particular subset of T cells that recognize lipids. And she's looking at crosstalk with dendritic cells, as well as tissue adaptation of these lipid-responsive NKT cells. And lastly, we have managed to recruit our chairwoman to the Crick, Carola Vinuesa, who's done seminal work on understanding follicular helper T cells, the role they play in germinal centers, as well as their regulation in germinal centers. And all this work has led to really important insight in the control or dysregulation that leads to diseases such as lupus. And here she has a big programmatic focus on functional rare low-frequency variants in lupus that contribute, and has managed to isolate these variants and set up specific mouse models that very nicely recapitulate aspects of these of this disease. And another focus she has is she's discovered that follicular regulatory T cells produce a neuronal protein neurotine, which is a very potent regulator of B cells, and in particular it limits the IgE class switch, so could be extremely important and potent in regulating asthma and allergy. And yes, finally, I just bring up my lab. I consider myself a lapsed immunologist. Our immune focus in the last years was very much on Th17 cells and the interesting plasticity in this T cell subset that either extended their repertoire of pro-inflammatory cytokines to play pathological roles in tissue autoimmune diseases such as EAE, or similarly a plasticity in the gut extended their repertoire to achieve more mucosal protective roles. But these days, my lab is very much focused on studying environmental influences, particularly in the gut, and the effect on regeneration by a transcription factor that is an environmental sensor, the aryl hydrocarbon receptor. And this was the Blitz tour. It was Blitz enough.
Thank you, Lisa. Given that you can talk to all of us in the break, we might just proceed to the last talk, who is given to us by Professor Paul Matthews from the Department of Brain Sciences at the Imperial College London. He's going to talk about immune mechanisms mediating genetic and environmental risks of neurodegeneration. Thank you.
Thank you. Let's see if this works. Well, almost. There. Good. So I'm going to speak to you about immune mechanisms that mediate genetic and environmental risks of neurodegeneration. But I'm going to speak about it in a very specific case to illustrate the general approach that we take. In the UK Dementia Research Institute Centre at Imperial College, we have about 6 researchers whose work to varying extents touches on immune regulation in the CNS niche. All of us are interested predominantly in the innate immune system. In the Department of Brain Sciences, there are others who are looking at the peculiar niches for adaptive immune system cells related to the brain in the subpile and perivascular regions in particular. I'll just focus on the first. What we're trying to understand in the context of Alzheimer's disease and Parkinson's primarily is the way in which environmental risk factors interact with genetics in order to produce disease. We envision this as a model, a very simple model, in which people inherit a genetic susceptibility to these diseases that is tipped over the edge and transition to the cellular phase of disease by environmental risk factors. I'm going to describe this in the particular instance of Alzheimer's disease, which is amongst the complex neurological diseases. There's this, we say, the sporadic disorders resulting from inheritance of multiple genetic risk factors, perhaps the one with the highest genetic risk load. There have been over 200 SNPs associated, 200 SNPs and potential genes or gene regulatory regions associated with Alzheimer's, of which now about 42 have very good data describing relationships between specific SNPs, mostly in regulatory regions, and individual genes. These genes allow us, give us a tool in which to anchor our interrogation of tissues in order to understand those factors that might be regulating, might be accounting for the environmental influence on the transition from susceptibility to early disease. There are a variety of approaches being used, but most of them now are settling on single nuclear or single cell analyses, which we can conduct on neurosurgical discard tissue in people who may not have, typically don't have overt signs of dementia, but as with many in the middle to early older age, may have the antecedents or form first of neurodegenerative diseases and Alzheimer's in later life, and from post-mortem tissue, where we can perform detailed single nuclear analyses. Here's one such, here's the outcome of one such analysis of a series of brains in which one can look at the cell type specificity for individual nuclei isolated post-mortem from rapidly frozen tissue, and use these to reconstruct the transcriptomes of the cells from which they were derived in order to understand the functional behaviors. What this allows us to do in the first instance is begin to zero in on those cells that may be most likely to be mediating this environment-gene interaction by looking for those cells that that express the highest load of genetic risk factors of those that we know. And the results here are really quite striking. As you know, we think Alzheimer's disease is traditionally envisioned as a neurodegenerative disease. I want you all as immunologists to leave today with the warm feeling that it's not, it's actually an immune disease, but an innate immune disease. Because if one looks at the enrichment of the genetic load for risk, in fact, this is using a magnum cell analysis in which the size of the bars expresses the relative genetic enrichment of cells in genes for risk. Those cells that are most enriched are not the neurons, which are at the lower part of the chart, where you see very small peaks, but actually the glial cells, and of the glial cells, overwhelmingly, the microglia. So this gives us a very specific target. And we're interested in looking, therefore, at the microglia, astrocyte, and endothelial interfaces, these special niches in the brain that logically also, based on what we know about their general functions, may mediate surveillance and interaction with the external environment. To analyze this in greater depth, what we've been doing most recently is enriching our single nuclear populations for the glial cells, and particularly for astrocytes and microglia, via transcriptional factor-directed FACS systems, which allow us to look at the individual cell types with considerable specificity. And most of those nuclei that we sequence, therefore, can come from the two cell populations of interest. From this, we can develop a rich understanding of the expression patterns in these cells, and develop, these are rather high-level co-expression maps in which each of the green spots that you see is an individual gene expressed in either microglia on your left or astrocytes on the right. Their relative spatial proximity is an expression of their degree of co-expression across a large population of cells, and highlighted in red are those specific spots that represent Alzheimer's risk genes. And what you can see is what I just described in more quantitative terms, that microglia and astrocytes, but microglia in particular, are highly enriched in these Alzheimer's risk products. Now, in fact, that map alone begins to give us some really interesting tools with which to dissect the cellular physiology of this disease. On the right is a crude cartoon of what we think we know about some of the early steps in the innate immune response in Alzheimer's disease, in which there's an interaction of lipids and amyloid protein, the causal agent in Alzheimer's disease, with external receptors that mediates an endosomal lysosomal pathway, resulting in contributing to clearance of the protein and to management of abnormal lipid load. You see the cartoon on the right. Now, in fact, if you look at that key stage of early invagination to endosomal transition, in fact, you see a cluster of genes, and that cluster of genes is co-localized within the co-expression map, suggesting that common regulons, or that they're all part of regulons, and common transcriptional mechanisms that are responsible for their regulation within the microglia. Now, in fact, we can learn more. We look at genes that are of particular interest to us, such as ApoE, which mediates the single largest genetic risk for Alzheimer's disease. We see the co-expression network shows us its nearest neighbors by co-expression, from which we can begin to make some crude inferences about functional roles on the left in microglia, on the right in astrocytes. And what you can see from the nearest neighbors, things like CD74, the complement pathway, TREM2, and so on, is that these are immune regulatory genes or immune response genes. If we look in the astrocytes, however, we see a quite different pattern. We see genes involved in amyloid processing, energy metabolism, early senescence, a quite mixed bag. We can take a further dive to begin to understand these patterns of transcriptional expression and what happens when we begin to activate microglia with, in the context of disease. If we look across people with Alzheimer's disease at a range of stages, or Alzheimer's pathology at a range of stages, we can look at the relationship between the local tissue amyloid load, the pathological protein driving the disease, and the gene expression patterns within, in this case, microglia. Now, I've simplified that by summarizing some of the pathways inferred from this gene expression pattern. And first, we see in the differential response model, which you see on the left in the panel, we see a number of pathways that we recognize as part of the probably more acute or early phase response of microglia to amyloid. We know this from studies in animal models conducted separately, those that are involved in amyloid clearance, in binding using scavenger receptors, in pathogen-related phagocytic pathways. But we also see a range of pathways that we also associate with the pro-inflammatory response, those NF-kappa B and key immune modulators like interleukin-4. So together, these tools help us begin to develop a detailed picture in the human tissue of what's happening across the broad chronic course of the disease, something we can't see in the mouse. What I haven't described is the way we can begin to start mapping these into individual niches using methods based on laser capture or biospatial transcriptomics. Several people in the group, notably Nathan Skeen, Sarah Marzi, Alexei Knott, who's just come from UCSF, Raffaella Nativio from Penn, are interested in understanding the epigenetic programs that drive these and how they in turn may be influenced by environmental signals. And Richard Reynolds and Paolo Marraro in the Department of Brain Sciences, in turn, in the context of chronic multiple sclerosis rather than Alzheimer's disease, are interested in the way the adaptive immune response interacts, particularly in subpile and perivascular niches. So I'm going to close just with a brief summary of what I've tried to tell you in giving you a small story from Alzheimer's disease to convince you that you're all neurodegeneration researchers, is that Alzheimer's risk genes suggest that the innate immune system is a main driver for the disease, that microglia play a central role. And I think how important it is to study the chronic human disease in human tissues, because they show a much broader, more diverse range of cell responses that are seen in the acute preclinical models. And we hope that, and in fact, they're already beginning to show us better ways of interpreting biomarkers, suggesting novel approaches for therapeutic proofs of principle, and in defining some new candidate genes. And thank you very much for your attention.
There is time for one or two questions max. Quick questions. Yes. Both. You're all right, Rob.
Hi, Paul. Very quickly, macroglia are clearly unique macrophages in the brain. They got a different ontology. How much do you think during the age there is a brain barrier breaking and there are monocytes replacing or taking the place? How much of this is going to be systemic, if you want, from actually a local macroglia problem?
Yes, I've greatly simplified the problem, obviously. So what it remains a major challenge to separate yolk sac-derived microglia from the myeloid cells that have macrophage-like characteristics. It is probably true that the perivascular macrophage pool is derived from the monocyte pool, and that the extent to which those traffic regularly is quite unknown, certainly unknown to me. And that is a very distinct group of the microglia macrophage population of the brain. I've skipped over that, but they have quite distinct transcriptomic properties, and in the context of Alzheimer's disease, behave in somewhat distinct ways. So I think they're potentially terribly important. And of course, I've skipped over the lymphocyte population in the brain, which is small but significant.
Thank you. Two very brief questions. One is, what's the evidence of peripheral monocytes can migrate into and integrate to become microglia-like? And the second question is, individuals who are immunodeficient or on chronic immunosuppression, what's the epidemiology for protection from Alzheimer's?
Right. So in the context of Alzheimer's disease, I don't have a good answer for you answer for your first question. There's been very little work done on this in animals, and what small work there has been suggests the trafficking is very limited. In the context of multiple sclerosis, which we understand better, it's clear that there is substantial trafficking of monocytes and transformation to macrophages, which are probably the major effectors of injury. And that the yolk sac-derived microglia then provide a primary neuroprotective downregulation of the inflammatory response. And it's an interesting model to think about carrying forward. And as we chatted earlier, I'm very intrigued that senescent cells in the CNS that are generated as a consequence of neurodegenerative pathways may trigger an adaptive immune response that includes monocyte trafficking. Now, the second question is about the epidemiology in the context of severe immunosuppression. I actually don't have those. I don't have those figures. Of course, what I can say is that in general, milder immunosuppression has not proven to be effective. And I think part of the clues to that are beginning to emerge as we understand the substantial repair properties of these immune cells. It is a complicated story, though, because in mice, the acute response, depending on the model, can go either way. So I personally believe that blunt immune suppression is probably not the answer, but there may be ways of enhancing certain classes of the immune response.
Hi all, so my name is James Harker. I'm going to chair this final short session. It's fantastic for those of you who've been able to stick with us either online or in person. For the final 3 talks is great, and first up we have Emma Davenport from the Sanger Institute, and Emma's going to share some of their work on immunogenetics.
Thank you very much. It's great to be here today, particularly to see so many people in person. And today I'm representing a number of groups at the Sanger Institute, and all of us, to some extent, have immunity and immunogenetics as an interest as part of our research. So we've got Rosa Venturtormo, who's in the cellular genetics program at Sanger, and she's really interested in tissue-resident immune cells. We've got Gosha Trinka, who has a focus on regulatory T cells and has recently started working on primary immunodeficiencies as one of her disease areas of interest. My team is really interested in systemic inflammation, so we work on sepsis and lupus. And in contrast to that, we have Carl Anderson, who is focused on inflammation in a particular organ, so he works on IBD. And then we have Nicole Soranzo, who's had long-term interest in the hematopoietic system. So today I'm just going to give you a snapshot of 3 projects that we have going on that sort of highlight some of the types of diseases that we're working on, the types of data that we're generating, and some of the approaches that we're taking in terms of the analysis. So some of the big themes that all of us at the Sanger who are involved in immune-related work, it really sort of covers how can we use genetic and transcriptomic data to understand heterogeneity within the disease. What role does genetic regulation play in terms of shaping the gene expression response to environmental perturbations? So that could be disease state stimulations or even treatments. And what are the genes, cell types, and pathways that mediate disease progression? So I think there's been quite a big shift in the field now, going from thinking about necessarily disease susceptibility to now thinking about within disease, what happens, why do some people sort of become sicker, whereas some people get better? So I'm going to start by just giving you a little overview of some work that I've been doing for a number of years with collaborators in Oxford, Barts and Imperial, and this is looking at the transcriptomic heterogeneity in response to sepsis. So sepsis is a life-threatening condition that occurs when the body's immune system is responding to an infection, but it ends up injuring its own tissues and organs. It's a very heterogeneous condition, and some of the challenges we face is there's no reliable diagnostic marker, there's no targeted treatment, and there's substantial variation in the individual response to sepsis. From previous work, we've identified 2 clusters from transcriptomic data. So we have an amazing cohort now of over 1,000 patients collected from the ICU with gene expression data, either microarray or RNA-seq. And we have 2 clusters within those patients. So the first of these is sepsis response signature 1, or SRS1, and the second is SRS2. And what we find is that SRS1 identifies a subgroup of individuals with an immunosuppressed phenotype and increased mortality. And so we're currently working on thinking about like the potential utility of having information about SRS phenotype, the SRS endotype when looking at these sepsis patients. And so obviously the main thing here is that it's a way of stratifying patients, so really highlighting those that have quite a specific immune response and a poor outcome. We're hoping that it's going to help us improve our understanding of the molecular mechanisms of disease and therefore lead to potentially new drug target identification, but also thinking about how this could be used as a biomarker, so in terms of disease progression or response to treatment. My team in particular, so as I said, this is a big collaboration and people have different areas that they're interested in, but my team in particular are interested in how the role that genetics is playing in the variation in the gene expression that we're observing. So we're trying to work out, like, are you predestined to go into one of these SRS groups? Do people move SRS group over time? Can we modulate it with treatment? And how much is the underlying DNA really sort of responsible for this difference in gene expression we see between these groups of people? One of the ways we're tackling this is through EQTL mapping. So this is where we look for an association between a genetic variant and then changes in the levels of gene expression. So for example, here we have a G allele in the promoter of a gene, and maybe a transcription factor binds more strongly. This results in more RNA being transcribed, and so you get this linear association between the genotype of your SNP and the expression of your gene. So you identify an EQTL. But we know EQTL can be context specific, and so we're particularly interested in looking for EQTL effects that are modulated by the environment. So in this case, EQTL effects that differ between whether an individual is in SRS1 or SRS2. So for example, if the transcription factor is not active in the SRS2 scenario, you won't get that increase in expression when there's a G allele, and you lose that EQTL effect. And so what we hope for here is that by identifying these EQTL interactions, it'll start telling us a little bit more about the mechanisms by which we're getting this variation in gene expression in the different groups, and potentially highlight some sort of key regulators in terms of the transcriptomic response. Some of the challenges with this work is it's all done on whole blood or PBMC, so mixed cell populations. And so something that all of us who are involved in immune research at Sanger are interested in is moving towards single-cell RNA sequencing to really get a better understanding of which cell types are contributing to the signals that we're observing in a lot of the studies we've previously conducted. So Sanger does science at scale, so of course that's what we have to do with these projects. But scale can mean very different things to different people at the institute. So for example, Rosa is very involved in cell atlasing, so for example the Human Cell Atlas project, and so for her scale means the number of cells that she's looking at, so she'll be going very, very deep and looking for rare cell types, but potentially only in very small numbers of people. At the other end of the extreme, we have Nicole's team, who won't be looking necessarily at many cells per person, but are looking at many, many individuals, and I'm just going to highlight one of her very exciting current projects. So this is Project Cardinal, and this is looking at the population scale single-cell RNA sequencing of PBMCs. So the aim is to profile blood cells from a multi-ethnic cohort. And their pilot study, let me just say pilot study, is to do 5,000 individuals from the UK Biobank and 1,500 individuals of Pakistani and Bangladeshi ancestry from the Genes and Health cohort. And so here they'll be doing 1,000 PBMCs on average per person, with the aim here of really comprehensively characterizing the blood cell types, the states and the ratios in diverse human populations, and creating an atlas of both cis and trans EQTL at the single cell level. So to be able to do this, a huge amount of infrastructure is currently having to go into place, because they're going to be collecting up to 80 fresh blood samples a day and shipping those overnight to the Institute to then do the PBMC isolation before loading onto the 10X to generate the single cell RNA-seq. So there's a potential to then increase this to the full UK Biobank, which is 50,000. So this is just unbelievable sort of quantities of data that could be generated. So obviously there's a huge amount going into making this as automated as possible. And so what we're hoping is that's then going to really change what's possible at the Sanger. And then other projects will be able to make use of the facilities and infrastructure. So for example, myself and Gosha are working with collaborators that you've already heard from this morning, so Marina, Jimmy Peters and Matthew Pickering, as well as our open targets industry partners, including GSK. And we are generating single-cell EQTL maps for both health and disease. So here we're looking at lupus and also healthy donors. And the aim here is to really try and identify the right cellular context in which immune disease variants are functioning. So we're going to do this by leveraging the single-cell RNA sequencing and generating single-cell EQTL mapping. So this is just a quick overview of the cohorts that we're going to be generating this data on. So as I mentioned, we're going to be focusing on lupus patients, and we're aiming to have equal numbers of individuals of Afro-Caribbean and European ancestry, and the same for the healthy donors. And we'll then be taking the PBMCs from those individuals, stimulating with 3 different stimuli and measuring the RNA, collecting RNA at 2 different time points to generate the single cell RNA sequencing. So this is going to enable us to do EQTL mapping across a range of different conditions, different time points, both disease agnostic in terms of the healthy donors and a systemic inflammatory disease in terms of lupus. And what we hope to get from this is information about the cell type in which the variant is potentially having a functional effect, the direction in which that effect is occurring, and really sort of help in terms of drug target identification and validation to sort of give a better idea of how to take this information further downstream for that. So that's quite a whistle-stop tour of just some of the big projects that we have going on at Sanger. Obviously, it hasn't covered everybody's research. We've got lots of exciting things going on, so please do get in contact with any of us if you think there are some interesting collaborative opportunities, and I'm happy to take any questions.
So are there any questions from the floor? I think that's a previous question for the previous speakers, so we might have to crowd applause and ask them that. So any questions from the floor? Maybe I can ask a really naive one, because obviously, as you said, it's science and scale, but everyone has a different view of what that scale is. How at Sanger do you decide if a project is 5,000 people and 1,000 cells, or 20,000 cells and 4 donors?
Yeah, I think it's really dependent on the question that we're trying to answer. And certainly one of the distinguishing features between Rosa, who's in cellular genetics, and Gosha and Carl and Nicole and myself who are in human genetics, is that we always want genetics at the heart of our experiments. So by definition, if we want to do things like EQTL mapping, we'll have to be having at least a couple of hundred individuals. So that's quite an easy distinction to make. Then obviously it gets very different depending on the cohort that you're working with, the availability of samples, how easy it is to get access to these projects. So yeah, it's very sort of hypothesis driven in terms of deciding what scale is appropriate.
Clearly impressive, super useful work, a really fantastic resource to have access to, and great that you guys are willing to participate in the network for sure.
Excellent. Thank you very much.
So there are more questions. I think the next 2 speakers are both online. So first up, we have Dr. Mark Peakman, who's here representing Sanofi. Hopefully it'll pop up on screen. Hi, Mark. Hopefully you can hear us.
I can hear you perfectly. Can you hear me?
Yes, I think so. Audience seems happy. Audience is happy. And Mark is going to tell us a little bit about the immunology and inflammation research that's going on at Sanofi. So over to you, Mark.
I'm assuming you can see my slides, James.
Yes, we can.
Okay, perfect. Well, first of all, thanks to Steve and colleagues for inviting us for this opportunity to tell you a little bit about what we're doing in Sanofi. So I'm here representing the therapeutic area in immunology and inflammation, and specifically its research branch. The research branch is led by John Burton, and you can see here that we divide ourselves into different clusters, each cluster being about 5 to 25 scientists with a cluster head. It just happens that on the left-hand side, those cluster heads are all ex-academics, so probably we're sitting in your seats one stage, I'm sure. And on the right-hand side, people have taken a more conventional career path through pharma and biotech. And we each lead an area of biology or disease that we're experts in or interested in. So we have type 2 inflammation, and I'll talk a little bit more about that. My own area is autoimmunity. We are very strongly underpinned by what we call precision immunology, and I'll touch upon that in a second. We have a complement cluster. Dupixent research cluster refers to a particular Sanofi drug, and I'll touch upon that in a couple of slides. And then type 1, type 17 inflammation, and a whole cluster on checkpoint biology. And these are the colleagues, and we work together. We have a lot of cross-fertilizing projects and synergistic work that leads us from early stage drug programs right through to development. And we probably have about 40 or 50 sort of programs, targets, modalities that we are pursuing at any one time. So I mentioned this drug Dupixent, and for those of you not familiar with it, I think it really has enabled Sanofi to gain a confidence that immunotherapy is really important for patients, can be really transformational, and therefore is something that is worth investing in at some scale. So Dupixent is a monoclonal antibody that blocks interleukin-4 receptor alpha. It was developed in a partnership between Sanofi and Regeneron, and it is turning to an absolute blockbuster drug for type 2 inflammation. In particular, at the moment, the main indications are asthma and atopic dermatitis. And by blockbuster, I mean that it's heading towards a sort of 10 billion per year income. And I'm not here to make that sound the important thing. I think what is important is that it really is transformational in these very difficult diseases. And I think what that's done for Sanofi, as I say, is to give them the confidence to think that they can now deepen that leadership in the type 2 space with other drugs that target other diseases or other aspects of the inflammatory cascade in type 2 immunity, including the examples that you see here, small molecules, bispecific antibodies, and also nanobodies, which I'll mention in a second. And again, that confidence then going towards the right to number 3 has enabled us to think about exploiting the same kinds of approaches in other disease areas. So going beyond type 2, as you saw in my previous slide. And so we are into what we call other immunology segments, whether they be innate inflammation around IRAK4 and RIP kinase, or anti-CD40 ligand, very focused on the adaptive immune system. And an increasing area of interest is the sort of counter-regulatory mechanisms that can be exploited around Treg biology. All of that activity is really strongly underpinned by a science base that we call precision immunology. It's led by Emanuele Di Rinaldis, and it's based on the premise that when you're dealing with complex diseases where you want to have multi-pathway targeting, that you need to understand the biology really well in order to forward translate, to build from the biology into the drug that you want to use in the right way. And that equally, when you have a drug that's successful, you can carry out what we call backward translation. And that's the example that I gave of the Dupixent research cluster, trying to understand what it means to be a responder, what it means to be a non-responder, what the different side effects might mean, and so on and so forth. And it gets back to this pretty standard sort of moniker for pharma of using selecting the right target, putting it in the right indication, with the right molecule, and treating the right patient. And that's something that I think we spend a lot of time thinking about and devote a lot of our effort to. Coupled with that, of course, to be successful in terms of immunotherapy, you need to have a reasonably comprehensive and broad arsenal. You need to be innovating all the time in that arsenal. And actually, I simplified this slide because there's probably about double the number of different modalities that we are currently investing in. But these are some of the ones that I'm going to pick out in some example areas in a couple of slides' time. So I mentioned already our ability to target multiple molecules through either bispecific and trispecific antibodies, or our nanobody platform, where we can couple specificities together into sort of chains of molecules that will target multiple inflammatory cytokines or receptors. So those are very much heading into the clinic now. We acquired an mRNA company and an mRNA delivery company that enables us to target mRNAs to single cell types earlier this year. We have this interesting expanded genetic alphabet opportunity to modify cytokines and other molecules, and I'll give you an example of that in a second. And then we're going beyond small molecule, sort of conventional approach of using small molecules to block, and morphing these into opportunities to degrade proteins, and I'll give you an example of that, as well as to covalently block protein function. And so I think, I mentioned that the clusters and the cluster leads have a strong desire to work together, because we recognize that there are pathway nodes that are going to be relevant across our disease areas of interest. And we also recognize, as I've mentioned in the last slide, that we have multiple opportunities for therapeutic modalities. And so it starts to become a fairly sort of organic structure in which we can be targeting different pathways for different diseases using different approaches. And as I mentioned, also beginning to explore the opportunity for doing that, exploiting what we now is an increasingly deep understanding of natural mechanisms of immunity. And I think, you know, the undruggable space is shrinking a little bit year by year as we get these better modalities and this better understanding of disease and disease pathways. So I said I would go into some examples in a little bit more depth, and that's what I'm going to do in the next 3 slides. So our desire to build on the success in type 2 inflammation is exemplified on this slide, where, you know, we've understood the biology of allergen responsiveness that includes epithelial responses to allergens through the production of TSLP, a very strong Th2 driving cytokine. And in that process, IL-13 is produced, and then IL-13 and TSLP really combine in a sort of inflammatory loop. And that loop can be interrupted potentially if one can develop a multi-specific mode of targeting these two simultaneously. And that's what we have done with our nanobody technology. And up in the right-hand corner, you can see an in vitro assay performed using tissues from humans, tissues from patients, in an allergen-driven chemokine assay. And you can see in the bottom right that the shift in response to the left-hand side of the lead nanobody targeting both TSLP and IL-13 compared with the single targeting comparators against TSLP or anti-IL-13 alone. So you would expect this single agent now to have really strong therapeutic benefits. And that's just gone into the clinic in phase 1 studies, and the lead indication there will be asthma. I also mentioned, you know, that we have a long-standing expertise in small molecules that we're now morphing into the degrader technology. We actually have I believe the first degrader to go into the clinic. It's an IRAK4 degrader, and it's based on the biology that you see in the center here, where IRAK4 occupies a pivotal position in the translation of pathogen receptors in the TLR family, driving type 1 interferon and inflammatory cytokine responses through activation of IRF5 and 7 and NF-kappa B. And the key thing about IRAK4 is that it has a kinase function which can be blocked conventionally using kinase inhibitors, but it also has a scaffold function, which could also be blocked by a conventional small molecule. But blocking either of those pathways will not give you complete limitation of IRAK4 activity. And so the degrader technologies that are beginning to emerge, it's a great opportunity now to see whether the degradation of IRAK4 is indeed an answer to drugging this pathway. And on the right-hand side, you can see that in these assays in human PBMCs stimulated through the TLR pathway, that again, we see a shift of responsiveness when we have the degrader compared to, for example, a kinase inhibitor. So this is a very exciting opportunity. It's been in phase 1 since March, and again, would go into those diseases where we believe through our precision immunology that there is a very strong driving element from these innate types of responses to pathogens. And then my final example slide is again something I mentioned, our increasing interest in natural immune networks, in this case, regulatory T cells biology. And here we acquired a company a couple of years ago called Synthorex. Synthorex are able to make cytokines and other molecules in which they have replaced the natural amino acids with synthetic amino acids. And in this case, that enables us to add a PEG molecule onto the cytokine interleukin-2. That doesn't just give you a different half-life and different pharmacology, but actually if you position the PEG properly or cleverly, in this case, you can make that IL-2 now a non-interleukin receptor beta binder, but a cytokine, an IL-2 that retains interaction with the interleukin-2 receptor alpha, and so has a favorable biology in that it will stimulate Tregs preferentially over conventional T cells. And in the top right, you see an in vivo study showing exactly that this interleukin-2 molecule can expand Tregs in the blood, in this case of non-human primates, really very reliably and dose-dependently without expanding NK cells or effector T cells. And in the lower right panel, you'll see that this cytokine will also limit inflammation in a delayed type hypersensitivity skin inflammation model, again in a dose-dependent fashion. So very exciting biology. This has just gone into the clinic as well, just started the phase 1 studies. We see this as an opportunity that can be applied to a range of autoimmune diseases where Tregs are known to be defective or dysfunctional. And perhaps getting to the end now, this is maybe the most important slide for this audience, because I think it shows the commitment of our therapeutic area to networking, to collaboration with other biotechs, with academic institutions, our desire to partner. And for example, I'm showing at the bottom right these two IMI, innovative medicine initiatives from the last few years. Inodia was at its time the largest IMI2 program to be awarded, and Sanofi is the lead pharma partner in Inodia. And on the right, 3TR actually trumped Inodia. It's about 80 million euros worth of funding to understand non-responsiveness to biologics across a range of different inflammatory and autoimmune disease. And again, Sanofi is the leader of that. And then perhaps of equal interest on the bottom right, we have a funding scheme for academic partnerships called the I Awards. Some of you may be familiar with this. I think if you feel you have areas of translational research interest that map onto the diseases and the biologies that I've mentioned, then it's certainly worth a conversation about the I Awards. They start off as about $100,000 of pump priming, but can then be converted into fairly serious money over a 2 or 3 year strategic research award. So that I think is something that people may want to follow up with. So to summarize, the therapeutic area is interested in understanding novel targets, pathways, molecules. All of that is our starting point for the programs that we put in place. We're keen to partner where we understand that there might be novel insights into patients, whether it be disease or responsive to therapy. We have a suite of novel technologies to bring to bear, and as I say, we are very keen to network and partner with academics and other entities alike. So, James, that's my last slide, and back to you.
This one works. Never mind. Next meeting.
I think we've lost Mark on audio.
Did you lose me completely throughout?
Yes, I think you lost us as well, maybe.
No, we heard your talk. We heard your talk.
Oh no, we heard your talk. Yeah.
Oh, okay.
Just did you hear Andreas's question?
No, I got complete silence at the end. Deafening
Did you
silence.
choose not to hear it? So I was just asking reminiscing about the anti-CD28 antibodies that turned out terrible in the end. I'm sure you guys must think very carefully how you actually get from non-human primates to humans, which in that specific case was really the point where things then failed. So I was just curious to hear what you do to kind of make sure that this is a safe transition. That was in reference to the IL-22, the modified IL-2.
Yes, I mean, you know, I've only been in the company a couple of years and came from academia where I was very aware of the pitfalls that you mentioned. I mean, what I see is a much greater emphasis on humanized models, and I think that is, and you know, other sophisticated ways of de-risking molecules as early as possible in terms of these potentially adverse effects. So,
yeah, so I was wondering this. I mean, did you, for instance, in this specific case, did you make a mouse that it was humanized for the IL-2 receptor, alpha, beta, and the common gamma chain to kind of see if all your predictions would pan out that way?
No, we didn't. But the molecule works in mouse, and the molecule works in primates, as you see. And I think with IL-2, you know, there's obviously 25 years of understanding of IL-2 in the human situation. So maybe that was something that we didn't have to worry about quite so much, although, you know, there can be some adverse biology. But I think as we go into new molecules, yes, absolutely, making knock-ins and so on is a sort of core part of the business, or humanizing animals.
Thank you.
I think thanks a lot, and thanks a lot, Mark, for joining us, and fantastic to have Sanofi as involved in the network. And I think with that, we'll move on to the last scientific speaker of today's. Oh, two more I've got, sorry. Can't count. So the next speaker is Professor Paul French from the Department of Physics. Paul is also online, I believe.
Can you hear me?
Paul, oh no.
Can you hear me?
Yeah.
Hello?
Yes, we can hear you.
Great.
And Paul's going to tell us about his multi-dimensional fluorescence imaging work.
Thank you. Good afternoon, ladies and gentlemen. I'm a member of the physics department in the photonics group, and I work very closely with Chris Dunsby, James McGinty, and Mark Neal. And together we run a group both at Imperial and also in a photonics satellite laboratory at the Crick. And we work on a range of mostly fluorescence imaging modalities, ranging from super resolved microscopy through to, I guess, conventional microscopy, high content analysis, but also implementing high content FLIM, FRET, and super resolved microscopy readouts in 2D cell culture and increasingly in 3D cell culture. We also work on disease models, I guess, C. elegans, zebrafish, and so on, mouse models, including with endoconfocal microscopy and sometimes clinical studies. And basically in this work, we are essentially using the same fluorescence technologies over again, implemented in different instruments. And a particular thing in our group that we've been working on for decades now is fluorescence lifetime imaging. This is where typically in fluorescence imaging, you label your protein or target of interest, and you put your excitation light on it, your fluorescent label lights up, and so you get the spatial distribution of your proteins, and if you can look over time and get the spatial temporal behavior. However, you can also do spectroscopy. You can look at spectral or lifetime readouts. I'm hearing some background noise. Is that a problem? No. You can get some spectral readouts such as fluorescence lifetime, which can tell you not just where is the labeled protein, but what's happening to it. And in particular, if you excite with a short pulse, you get a signal which is independent of the concentration, it's independent of attenuation in the sample. If it's quantitative, you can account for autofluorescence. And so it gives you quantitative readouts, particularly for FRET, force to resonant energy transfer, which you can use to probe protein interactions, drug target engagement, etc. And we've implemented this in a range of systems, including in endoscopes and multi-well plate readers. I'll give you a couple of examples. And so, as I'm sure you know better than me, when you're interested in FRET for looking at protein-protein interactions or drug target engagement, you typically label your two interacting parties with a donor and acceptor fluorophore. And if they come together, you get an energy transfer. So the donor fluorophore gets dimmer and the acceptor fluorophore gets brighter. And this energy transfer is a new decay mechanism for the donor, and so it loses its energy faster, and this results in a decreasing fluorescence lifetime. And if we measure this carefully, we can first of all detect interaction, but we can also quantify it. We can calculate the population fractions, we can calculate the FRET efficiency and the dissociation constants of interactions. So I'm going to give you one example from work done with Eric Sahai at the Crick, where we implemented this in a confocal film endomicroscope. So this was the Morna-K endomicroscope, which we modified to do fluorescence lifetime imaging using time-correlated single photon counting. This gives you an example of the kind of image that we get through the endoscope, single cell resolved FRET readouts. And this is a comparable image of the same sample. And this is a mouse model where these cells are expressing an ERK-FRET biosensor. And you can see that the endoscope isn't such high resolution, but nevertheless, we still get single cell resolved readouts, and the endoscope tip is 2 millimeters thick. The microscope objective is rather wider. And if you want to look inside a mouse, it's clear which one is preferable. One of the challenges of in vivo imaging is, of course, that the animal is alive and it breathes. And so this causes all sorts of motion artifacts, but we can tidy that up, sort it out, get quantitative per cell readouts. And in the study that we did with Eric, we were looking at the drug target engagement. We were looking at doxorubicin binding its target, intercalating with DNA. And Eric had generated a mouse model in which the histones were labeled with GFP. It turns out that doxorubicin is fluorescent and can act as an acceptor to the GFP donor, so that when doxorubicin binds its target, you get FRET and lifetime quenching of the GFP. So by reading out the decrease in fluorescence lifetime, we can directly measure the amount of doxorubicin binding the target, single cell resolved in vivo. And for example, we're able to plot histograms on a per cell basis of the population fraction of GFP molecules that are bound to doxorubicin, and therefore we can calculate the intracellular concentration of doxorubicin. And what you see here is this is a control, so no doxorubicin, so we have a low population fraction, very low intracellular concentration. Here we have the doxorubicin concentration for an intraperitoneal injection of doxorubicin into the mouse, or an intravenous injection, intraperitoneal injection or intravenous injection. And what you can see here is that the drug target engagement is much higher for the intraperitoneal injection. And so this typically lets you separate the, if you see the therapy fails, you can separate whether it's due to the drug binding its target or due to the subsequent mechanism. So we can directly measure and quantify drug target engagement and start to study drug resistance mechanisms in vivo. We could use something like this to look at different drug delivery approaches. And this is a drug target engagement. We could also look at this, use this to study protein-protein interactions in vivo or a FRET biosensor in vivo. So that's endoscopy, which is relatively low throughput. We've also tried to make this technology high throughput, and we've built multi-well plate readers where typically we can acquire FLIM-FRET data in 10 seconds per field of view, which means we can take one field of view per well in a 96 well plate in 960 seconds, which means in an afternoon's experiment, we get data from thousands of cells, thousands of fields of view, and hundreds of fields of view and thousands of cells. And this means we have much more robust data than you normally associate with fluorescence lifetime imaging. We've created an open source instrument for this, which you can share. And I'll just give you one example. We did a study with Pfizer where they were interested in HIV gag oligomerization as part of the HIV cycle. So in HIV, when the virus infects the cell, hijacks the cellular mechanisms, and it creates these new virus-like particles, which are formed of a shell of gag protein. Pfizer were interested to see, could we get a readout of that? It turns out you can have a good model of that simply by transfecting gag protein into cells, and these go to the surface and form these virus-like particles. And if we stochastically label the gag protein with YFP and CFP, when these virus-like particles are formed, you see FRET between the CFP and YFP. And so this is a readout of the formation of the particles. In order for these particles to form, it's necessary that endogenous NMT enzymes add a myristic acid moiety to the N-terminus of the GAG protein, and my colleague Ed Tate at Imperial has developed inhibitors of this process, so we can use this to test inhibitors. So if we add the inhibitor and it works, we see low fret because we don't get the virus-like particles. If the inhibitor fails to work, we see high fret. And it also turns out that CFP is unusually can fret with itself, and you get a decrease in fluorescence lifetime. And so this is quite a nice readout, so we can do the same experiment with that. And in this simple screen that we did, this is a multi-well plate where we have a map of inhibitor concentration, and this is a heat map calculated from the per cell fluorescence lifetime readouts, which gives us the lifetime here as a readout of fret efficiency. And then we can plot these dose response curves of inhibitor and for both homofret and heterofret, and we can, for example, get a reasonable Z prime to do quantitative assays. So in another project with Eric Sahai, we're interested in cancer, studying drug resistance, and Alex de Mauer is primarily working on this. And here I'm showing you some movies, not lifetime, but spectral fret this time, of a body of cells which have been treated with a cancer drug. And in general, there's a response in the ERK pathway, and so you get a change of the readout of the ERK biosensor. But what you can see here is a small number of cells give a different response, and a small number of cells here give a different response. And so we've developed software tools, integrating open source tools like TrackMake to track the individual trajectory of every cell, correlate it with the readout of response according to the ERK biosensor, and Alex is studying the response of these cells as a function of where the interaction comes in the cell cycle. And this is just one example of the kind of study that you can undertake. Now, this work is challenging because these experiments typically take tens of hours, and when you're doing fluorescence assays over tens of hours, you have problems with phototoxicity. But on the other hand, if you don't continuously monitor the cells, it's hard to track the individual trajectory and the mitosis. And so you really would like a low phototoxic way of monitoring the cells. And we believe we can do that with phase contrast microscopy, not using fluorescence at all. And we recently developed a single shot differential phase contrast microscopy technique, which essentially acquires a set of images where, I won't go into the details, but basically you put these different masks in the back focal plane, you acquire 4 images, and from these you can calculate quantitative phase contrast images. We found a way to do that with a novel polarization resolving camera so that we can take 4 images in a single shot. So we acquire these 4 images, and from these we calculate these phase contrast images. These are quantitative. This is an example of the profile and so on, as you can see here. And we can already use these now for single cell tracking and segmentation. We can use these, for example, to provide masks to bin the photons from cells to get more quantitative fret readout responses. And because this is really fast, we can apply it to dynamic subjects. This is applying it to a C. elegans. Here you see the live C. elegans, and if you look here, I think there's something called the pharynx, which is the C. elegans digesting some food. And because we're using polarization as a way of getting this readout signal, it doesn't matter what wavelength radiation we can use. We can use a low phototoxicity infrared radiation, but also we can easily use it separate spectral channels for fluorescence. And so this is an overlay of fluorescence imaging with the endothelial, with these cells labeled with GFP, and the red channel is showing the phase contrast. And we're going to use this, for example, to study models of infection in C. elegans. And again, just to show the speed of this technique, this is a movie of water droplets evaporating on a glass slide. And essentially, this shows that we can get quantitative and morphological information on a fast time scale, and then we can sample fluorescence readouts on a slower time scale and avoid phototoxicity. And so this question of looking at the heterogeneity in response to drugs is obviously of interest if you're studying drug resistance. And at the moment, that project is looking at 2D cell culture. But of course, there's a great deal of interest in 3D cell culture. And we have a significant project developing high content analysis platforms where we're imaging 3D cell cultures, spheroids and patient-derived organoids, where we want to develop this at throughput. And we're particularly using a technique called oblique plane microscopy, which is a form of very fast light sheet microscopy developed by my colleague Chris Dunsby. We're also using a spinning disk fluorescence lifetime imaging platform, and we're developing a multi-beam, multi-photon, multi-well plate platform for imaging thicker tissue samples. This is part of a CI UK accelerator, for which the partners include the Crick, the Institute of Research in Barcelona, the Institute of Cancer Research, and the CI UK Centre in Edinburgh. And just to give you show you some examples of the kind of data that you can get from oblique plane microscopy. You can image very fast at up to greater than 25 volumes a second. So Chris has done studies of live cardiomyocytes looking at calcium sparks. You can also have your light sheet coming out of your objective, slide a multi-well plate through it and image the organoids. And very recently, Chris published a new system in collaboration with investigators at the Crick, which accelerates, which improves the resolution. And I'll just show you a few examples of the kind of 3D data that you can acquire in multi-well plate arrays of patient-derived organoids or other tissue cultures. This is an ERK biosensor expressed in mutant mouse lung tissue treated with inhibitors. This is looking at stem cell division and proliferation markers in patient-derived organoids from Batel's group in Barcelona. This is work on melanoma spheroids from Chris Bakel's group at the ICR. And this is an example of a 3D phenotypic assay of glioma cells near a hypoxic core in glioma stem cell patient-derived organoids. And so all of these things, they're not just imaged one-off. These are imaged at speed in 96 well plates. So in a time-lapse assay, and we're now running these over multiple days, you can image 30 or 40 organoids continuously. And then rapidly moving on, we're also working on super resolve microscopy. We developed recently a robust form of super resolve microscopy based on single molecule localization, a technique we call EasyStorm. This is an example working with Louise Donnelly and Peter Barnes in NHLI, where we're interested in studying defective phagocytosis. And this is a super resolved image of bacteria in THP1 cells, which serve as a model for macrophage. And basically, you can see that the storm image is much more quantitative and the quantitation will be much better looking at the bacterial load compared to the wide field image. This is a low cost technique, and so we're translating this to partners in India and other labs that want to access super resolve microscopy at low cost. We're developing modular technologies that means that we can replicate these systems, and that's important not just for widening access but also for increasing throughput, because if you want to go faster, and super resolve microscopy is usually thought of as pretty slow, one way to go faster is to have 10 microscopes. So if you can have 10 microscopes that cost if each microscope costs a tenth of the commercial instrument, then scaling is quite practical. But we can also go faster through automation. But before I get to that, I want to present a very recent result that we've developed with Terry King and Candice Rufos at Hammersmith Charing Cross Hospital. So they are renal pathologists, and their interest, and typically in renal pathology in the NHS, one uses electron microscopy to diagnose kidney disease, particularly looking at structures like the glomerulus. And what you see here are wide field images and super resolved images at different zooms. And you can see that in the super resolved images, for example, E and F, we're seeing structures that we can't really quantify in the wide field images, but we can directly correlate them to the images that we see in electron microscopy. And another example would be measuring the thickness of the glomerular basement membrane, which is a key diagnostic marker. If we look in an epifluorescence or immunofluorescence microscope, we measure over 650 nanometers, but we can get a much more realistic thickness of 212 nanometers using this super resolved microscopy technique. So if we can make this low cost, we can really have an instrument that would be affordable in the NHS, and we can build this microscope for about £20,000. And NHS electron microscopy is under pressure. I think more than half the centres have closed, and there's a shortage of the expert technical staff to run such instruments. This technique is akin to immunofluorescence. It's using standard immunoantibodies, and so this could, I hope, find wide application. And as I said, we can also do this in high throughput. So we're building multi-well plates, and the goal here is to build high throughput automated assays utilizing this higher resolution. This is another collaboration with Chris Baker at the ICR, where we've labeled actin and paxillin. He's looking at focal adhesions. These are epifluorescence images and storm images from the 96 well plate. These are zoomed in regions of interest. And now we have a vast amount of super resolved data, and we're trying to develop a pipeline so that we can extract quantitative parameters to quantify, for example, what happens to these focal adhesions as we treat them with drugs or knockout. Have I run out of time?
We've run out of time, if that's okay.
I had one more slide.
Show the last slide.
Well,
we'll skip questions.
Here's my last slide. We can also image larger volumes optical tomography, and this is 3D cell masses, for example, in study of diabetes, where we're quantifying beta cells. This is an example of looking at cleared tissue, and this is an example of looking at live zebrafish, both embryos and adult zebrafish. This is a zebrafish with a GFP labeled tumor. So I will stop there. Thank you very much.
Oh, absolutely amazing. Absolutely amazing imaging, absolutely amazing technical development. I think we all know who we're going to go to for our kind of when we want to visualize the immune system and try and understand pharmacokinetics. So thank you so much for your presentation. If anyone does have questions, maybe message or email Paul. And this time I'm going to get it right. Our final scientific speaker.
Yeah, I'm sorry to disappoint you. That was another one. It's me, so I'll be quick.
It's Professor Ian McNeish, who's here representing the Department of Surgery and Cancer. So over to Ian.
Thanks. I'm aware I'm the last thing, more or less, between you and a drink, so I'll make it swift. So I'm Ian McNeish from Imperial. I'm going to talk a bit about what we do in solid tumour oncology at Imperial. Marina will have told you about hemato-oncology. And I'll give you a couple of examples. Inevitably, we're running thousands of trials of immune checkpoint inhibitors, but leading in hepatocellular carcinoma and de-escalation of non-small cell lung cancer. Hector Kuhn interested in immunometabolism, and with Julian Marchesi, lots of work looking at the gut microbiome. But I just thought I'd talk about two specific projects to give you an idea of what we're doing in ovarian cancer, which is my cancer. It is actually the most interesting cancer in the whole wide world, just in case you were worried. And it presents a huge challenge, but it's a disease of utter chaos. It is genomically the most abnormal and hard to decipher cancer of all the solid malignancies, and poses huge challenges for us. But we've known for about 20 years, since George Kukos showed us that the presence of intratumoral CD8 positive T cells is incredibly strongly prognostic, but we still haven't a clue why that is or what's driving that. And more to the point, you can give hundreds and hundreds of women immune checkpoint inhibitors, as we have done in a series of not terribly well outthought clinical trials, and we make absolutely no impact on them whatsoever. So my lab's really been interested in trying to understand what's driving the immune microenvironment environment in ovarian cancer. And being a medic, I think rather simplistically and stupidly. So my statements are the blindingly obvious in cancer. Are that you can't have an immune microtumor environment without a tumor. So there must be something about the tumor that is driving the immune response, particularly in ovarian cancer, which tends to live entirely within the peritoneal cavity, which I'm sure some of you know is a very interesting and unusual immunological space. And therefore, specific mutations, mutational processes or signatures, or indeed epigenomic states, must correlate with immunogenicity. Wherever possible, we try to use primary patient material. My surgeons, top right, are fantastic. They give me tumors by the kilogram momentum. And so we're doing lots and lots of primary ex vivo culture in collaboration with Darryl Overby in bioengineering. We're also working with the Sanger and indeed Paul French, looking at primary ascites. So ovarian cancer patients develop ascites, which is full of every immune cell you could be interested in. and we've developed really good techniques now for single and multi-cell organoid culture. But the workhorse, or the work mouse in my lab, is this ID8 model that we developed, an allelic series whereby we can recreate individual mutations in high-grade ovarian cancer one by one to address the question of how do individual mutations drive different immune microenvironments in ovarian cancer. Sarah Spear, a great postdoc in my lab, has been looking at loss of P10, which is prognostically very poor in humans and indeed very poor in our mouse models. And she's shown that just the loss of P10 significantly alters immune microenvironment, significantly more tissue-resident macrophages, probably fewer monocyte-derived, and definitely many fewer CD3, but mainly CD8 positive cells. And you can reverse that by adding back in BRCA2. So two single genes altered, significantly altering the immune microenvironment. And when you look specifically at macrophages, P10 drives the expansion of unique macrophage populations in this single cell RNA suite in this cluster 2, driven apparently by HEMOX1 expression. So this is really exciting, looking at how individual genes can change the immune microenvironment in ovarian cancer. The other thing we're looking at is epigenetics. Lots of interest in epigenetics at Imperial. You'll be aware that Wei-Ping Zhou a few years ago looked at DNA methyltransferase inhibitors as a way of reactivating Th1 chemokines. We've been interested not in DNA methyltransferase inhibitors, but histone lysine methyltransferase inhibitors, in particular drugs targeting EHMT2 and EZH2. Partially driven by Matt Fuchter, whom I think you heard from earlier on today when I was in clinic, who's developed the first-in-class dual inhibitor of EZH2 and EHMT2. And we've been working on that recently, showing on the right that if you put that on our mouse and indeed human ovarian cancer cells, you see dramatic upregulation of chemokines. There it is in our primary cells from 7 of my patients. The RNA-seq, all the gene expression pathways are immune-related and largely innate immune-related, and defense response to virus. That's correlated perfectly with the ATAC-seq, looking at change in chromatin structure. Again, all the genes associated with that are immunological and innate, defense response to virus. When we do multi-parameter flow, we see significant changes in NK cells, T-cell match, CD8 cell maturation, significant upregulation of granzyme B, and marked upregulation of chemokine receptor CXL3 in particular in multiple different anatomical locations in multiple different immune cell subtypes. We also see changes in myeloid populations, including a reduction in immature dendritic cells. So what is going on? There's multiple things that could be happening when you start altering the epigenome in cancer. We know that there are marked changes in the epigenome in ovarian cancer, especially as it evolves and becomes resistant to chemotherapy. But one thing, driven mainly by those RNA-seq data, looking at innate immune responses, and in particular, apparent viral response pathways, was looking at endogenous retroviruses and other transposable elements. Indeed, Steve Balin suggested this a couple of years ago in Kate Chapanelli's paper in Cancer Cell. So if you inhibit EZH2 and G9A in human ovarian cancer cell lines, again, you see marked upregulation of defense response to virus type gene expression signatures. And if you look particularly at transposable elements, you see high degrees of upregulation, in particular endogenous retroviruses that are mainly of the ERV1 class. We also see that in mouse. So these are my mouse cells, p53 null. And again, we're seeing marked upregulation of endogenous retroviruses, both in cells and in the tumors in the mice following treatment. And reassuringly and gratifyingly, this drug makes mice live longer. It's a particularly aggressive model of high-grade ovarian cancer, but we get significant extension of survival, reductions in tumor volumes, and absence of ascites at concentrations and doses that are non-cytotoxic. So we believe that by altering chromatin state in tumor cells, that we're clearly driving probably an antiviral response driven potentially by transposable elements and ERVs that is enhancing the microenvironment in our high-grade ovarian cancer. Where do we go with this next? Clearly, we're working towards a phase 1 trial. We're doing preclinical tox models, but clearly there's lots of interesting immunology that we're doing at the moment. But I just wanted gratuitously to steal a slide that Alan Melcher, I think, showed you earlier on today. And I think Alan said, we don't know anything, and there's a huge amount of cancer immunology that we don't know. Sadly, that hasn't stopped us running hundreds of clinical trials. Universally in ovarian cancer, so far, they have been embarrassingly negative. And so my plea to everybody here, I think is probably the same as Alan's plea, is that we really need to integrate basic translational and clinical aspects of our immunology if we're really going to make meaningful improvements in cancers other than melanoma and non-small cell lung cancer. And obviously, I have a vested interest in trying to make this work in ovarian cancer, where, as I said, we've not had much success so far. So I told you I'd be quick. Thank you very much indeed. And I'll hand you over to Steve before we all have a drink. Thank you.
Does anyone have, oh no, Ian's off. Does anyone have a pressing question? Otherwise, it sounds like Ian might be available outside for to share a question over a beverage.
How about that? Thanks.
So over to Steve for the closeout. Possible.
That's not going to work. Right. There we go. Let me get that to come up on the screen.
Is it plugged in the back?
I don't know. No idea. That would help. Someone going to come and help us.
Can we get some assistance at the front?
Hey. Right. Thanks. I don't know anyone who did that, but anyway. Okay, so hope you've enjoyed listening to all these great talks during the day. It gives you a really good idea of all the different things that are going to be going on in the network, and hopefully you might have already had some thoughts about new collaborations that you might want to start. I certainly have. Saw TPL2 coming up in one of the lists that Paul Matthews put up, so I'm very excited about that. The next thing that's going to happen in the network that I've already mentioned is the first workshop, which is on March the 4th, and that's going to be focusing on cancer immunology and being run by Alan Melchers and Jessica Stridd, and that'll be taking place here. So you may want to either turn up in person or online to listen to one of the 8 talks that are going to be given at that meeting. And also the Journal Club will be starting in January. And I'm not going to say anything else apart from to say that in a year's time, we're going to be back again for the 3rd London Infections and Immunity Symposium. We've already got all the speakers lined up for this. Keynote speakers, Dorian Cantrell and Gordon Brown, and a whole load of people from the Crick, Imperial College, University College and King's. I've also invited each of the companies that are taking part in today's talk if they want to give a presentation, and
a couple have already agreed to that. I only had that idea a couple of days ago, so I'm waiting for replies from a couple of the ones that haven't yet responded. Hopefully we'll see you at that. And that's going to be a basic research symposium that's going to be rather than an overview type thing like today. So hopefully you can stay around and have a bit of food and drink outside. Thanks very much.